Hello and welcome everyone to our second and final day of the virtual 2021 Fall ADRC meeting. I'm Hannah Rosentrader and I'm a manager of special projects here at the National Alzheimer's Coordinating Center. And I just wanted to thank you all for joining us early on the West Coast or right in the middle of your second cup of coffee on the East Coast for our highly anticipated director's session happening today from 7 a.m. to 11 a.m. Pacific time. After I introduce Dr. Oscar Lopez, Director of the University of Pittsburgh ADRC and incoming chair of the ADRC Executive Committee. Welcome, Oscar, to our virtual stage, and you can get things started for us.
Thank you, Hannah. And thank you for your introduction. I'm the director of the University of Pittsburgh ADRC, and I'm taking over the Directors Executive Committee from Andy Sagan, who did a wonderful job in previous meetings. It is very unfortunate that we cannot meet in person this time. I hope that our next meeting will be in person and the virus would be part of the history of our country and of our world. I would like to thank Dr. Silverberg, Elliott, and Donnelly for providing the framework for this meeting. And Hannah Rosentrader from the NACC has done a wonderful job coordinating the presentations. I would like to welcome our two new centers, the South Texas ADRC, comprised by the University of Texas Health Science Center at San Antonio, and the University of Texas Rio Grande Valley, led by Dr. Sudha Seshadri and Gladys Mestri, and the Duke and University of North Carolina ADRC, which is led by Dr. Heather Wilson and Gwen Garden. We have a busy morning with many interesting presentations. We'll initiate the morning with an NIA update from Dr. Hodes, Masliah, and Silverberg. Dr. Kukull will provide us with an update from the NACC. This will be followed by the science presentations. The first presentation will be from Angela Taylor from the Lewy Body Dementia Association. She will present the Nomenclature Initiative. Dr. Petersen from Mayo Clinic and Dr. Weintraub from Northwestern will be in the virtual platform, as well as Jason Karlowicz. It's important to note that Dr. Petersen and Angela Taylor are co-chairs of the steering committee. Dr. Karlowicz chairs the Public Stakeholder Working Group, and Marwan Sabak chairs the Clinical Care Working Group. And Dr. Weintraub chairs the Research Working Group. This will be followed by Dr. Paul Thompson from USC, who will present the ENIGMA Project. This is the Enhancing Neuroimaging Genetics through Meta-Analysis. This is a worldwide consortium of researchers led by Dr. Paul Thompson at USC, dedicated to identify the genetic basis of brain structure and function. This will follow by Dr. Singleton from the NIH, who will present an overview of the NIH Intramural Center for Alzheimer's Disease and Related Dementias, the CARD. And finally, Dr. Thomas Karikari from University of Gothenburg, in his transition to the University of Pittsburgh, will give us an update on blood biomarkers. This is a hot topic today, and Dr. Karikari will provide us with the experience of a very active center involved in the development of blood biomarkers for multiple neurodegenerative disorders. So the next speaker will be Dr. Hodes. Dr. Hodes.
Thank you, Oscar. It's a pleasure to be here with you virtually. Wish we were in person again, but as you've heard from all of us, hope to be together again in person soon. There are a number of updates I'd like to share with you in the presentation now. And next slide, Nina. First, as each year, budget and spending, give you an update of where we are, what we know, and of course, what we don't yet know about our budget. Next, please. So as we have begun this week, fiscal FY22, this is the status of the appropriations process. Back in May, the president proposed a total of $51 billion for NIH, which would be a $9 billion increase. And notably, this included within that $9 billion, $6.5 billion to establish so-called ARPA-H, the Advanced Research Project Agencies for Health. Now, the language noted that this would focus on cancer and other diseases such as diabetes and Alzheimer's. But in fact, although it's a long way to go till we have ARPA and understand its full scope, it's not likely to be in any way principal focus upon specific diseases, but rather on the broad structures that will serve these diseases and others through such things as broad molecular atlases or ways in which to mine and analyze data across broad populations. That was the president's budget in May, and then Senate and House appropriation bills moved to the next step. In July, the House passed its labor HHS appropriations bill, which provided for $49.4 billion for NIH. That was an increase of $7 billion. Within this, $3 billion for ARPA-H, and importantly for NIA, $4.258 billion, which would include a $200 million increase for AD/ADRD above current levels. We're still awaiting release from the Senate, which is expected in the next week or two, and then we'll work from there. Right now, we are functioning under a continuing resolution, which many of you will understand and be used to. This is through December 3rd, happily keeps the government in business till that time, with funding at a level equivalent to what we had last year in FY21. So it'll be at least until that date, December 3rd, as practice would indicate, before we know what our budget will be for FY22. Next, please. If we look back over the remarkable change of these past years with increased funding, bipartisan, bicameral support for AD and ADRD research, if we look at the change from FY 2015 through FY 2020, that's the last year in which we have complete data to analyze, there's been a 4.5 to 5-fold increase in the various components of AD and ADRD spending. Extraordinary, certainly far in excess of any of the other areas of NIH, and truly historic. And it's a testimony to the work that's gone on in the centers and elsewhere, and the confidence consequent to that Congress and appropriators and administrations have had that have led to this increase. Next, please. This is to give you an idea. Each year, if we look at the competing awards issued in each year, how they break down in these bars by color categories for dementia care, population studies, clinical interventions, diagnosis, molecular pathogenesis. You can see the distribution. It varies some from year to year, but it's a balance each year across all of these categories. Next, please. In terms of pay lines for FY 2021, the year just passed, the general pay line, which is for research supported by NIA other than Alzheimer's and Alzheimer's-related dementias, the 10th percentile for the most common under $500,000 applications, clearly far less than is sufficient to fund the outstanding research we have in those areas. Advantages given, as always, to NI, new investigators, and ESIs, early stage investigators. Fortunately for AD/ADRD, we're able to maintain the pay lines as in previous years at the 28th percentile for 31st and 33rd, again encouraging new and early stage investigators. And if we look at other mechanisms on the next slide, and focus for AD/ADRD again for program projects, a priority score of 40 was the funding threshold. For other NIA reviewed research, similarly for career development or K awards, 35, and fellowships, 40. Gratifying that we're able to maintain these levels of support for the many outstanding applications that we are receiving. Next, please. Now turn to the bypass budget exercise, which in this past year was the bypass budget for FY 2023. Next slide, please. As a reminder, the bypass budget language derived from a congressional act which required that each fiscal year through 2025 that the director of NIH, so that's currently Francis Collins, prepare and submit directly to the president for review and transmittal to Congress, opportunity for comment, but importantly without change. So the NIH bypass budget, professional judgment budget, goes to Congress to indicate the increase in funding required to optimally pursue the goals of the national plan. Next slide, please. This is distributed in milestones across 8 cadre categories, which are the common Alzheimer's disease research ontology that was originated collaboratively by NIA and the Alzheimer's Association to help to categorize research. It also provides the framework for IDROP, the International Disease Research Portfolio, which allows tracking and implementation in all these budget areas over the years and across global funders. Next slide, please. So in this FY 2023 budget, which was released in July of this year, what you can see listed here are the amount of increase required in each of the categories noted. These are increases above the FY 2021 level of $3.2 billion for Alzheimer's disease. The amounts over that in each of these categories necessary to pursue with optimal efficiency research across the areas of AD and ADRD. The total or sum of these was $376 million. But of course, now that we've been functioning for a number of years on these increasing appropriations, there are funds that become available for completed projects that's now available for application to these new priorities. That was a total of $150 million. And so the total request is the difference there. So next slide, please. Some $226 million summarized here. If that is added to the $3.2 billion 21 enacted level, that would propose for FY 2023 proposed resources needed of $3.4 billion for AD/ADRD research. And of course, we await the appropriation for FY 22 to see what happens there, which will of course influence, in fact, the real implementation plans for FY 23. Next, please. So I'd emphasize that in addition to the numbers, the bypass budget includes a narrative, which this year in particular was a very gratifying and impressive accounting of AD and ADRD progress over past year. So I recommend to any of you, please to take a look, examples of science advances across multiple research topics meant to inform all audiences, importantly, including the congressional audiences, and to influence by continued support, we hope, their appropriations enactment. The link is there for you to see the full narrative. Next, please. In terms of implementation tracking, you can look in iDrop with the web-based tools we have here to look at the progress that is detailed milestone by milestone as research continues. This links, therefore, to the bypass budgets and provides to all audiences, including the congressional funders themselves, with an ability to track precisely what it is being accomplished over time as a result of the appropriations. Next, please. Working towards health equity. This is a theme that has engaged all of us increasingly over these past years. Next slide, please. And at NIH, it is embodied in the UNITE initiative, reflecting our commitment to ending structural racisms. You can see the acronym there, U for understanding, and importantly for new research on health disparities, minority and health equities, largely the extramural research community. And I would stress here, most importantly and germane to this meeting, very important to continue the outstanding work that's been done in AD and ADRD to look at diversity, disparities, and equity, the factors that contribute to differential susceptibility to dementias, and to the differential approaches to their treatment, prevention, and care. We've learned a great deal by looking at diverse populations and by serving them. And I think that Alzheimer's research should continue the important and impressive tradition it has had, and increases in current and proposed initiatives to support this overarching NIH commitment. Next slide, please. One of the initiatives that you've been hearing about, I think, is the CROMS initiative, the Clinical Research Operating Management System. This reflects the fact that, like many at NIH, we've been challenged in providing the kind of detailed tracking for clinical research, including clinical trials, that's necessary to judge, progress, and feedback to inform these studies in terms of their recruitment, retention, and importantly, the reflection of the diversity in populations trying to achieve. Currently, through the initial phases, it's anticipated that by the end of the year, we hope to have incorporated a good deal of the AD/ADRD clinical trials. Eventually, it'll be available to all NIA projects supporting human subject research. We think it will, once instituted, actually enhance and facilitate the burden for investigators and investigator teams in tracking their recruitment, retention, and the characteristics of their populations. Next, please. The challenge of recruitment, including recruitment of diverse populations, has been a theme we've been working with the centers on, with Alzheimer's Association, with other advocate groups over these past years. And illustrated here are two initiatives to that end. ADORE, the Alzheimer's Dementia Outreach, Recruitment, and Engagement function, which online provides a database of accumulating resources for effective means for recruitment. And related to that, Outreach Pro, which provides tools for potentially tailored approaches that can be taken by individual trial recruitment for accomplishing those ends. Next, please. There is another initiative that is under discussion, was the topic of a seminar back in April and continues to be an ongoing conversation. And that's the potential for establishing a practice-based research network in order to bring closer to the practice communities the research infrastructure and to facilitate the recruitment, again, of diverse populations geographically as well as racial, ethnically, into clinical research. So we'll look forward to, with you, working on this potential approach in which the centers may play a very important and central role. Next, please. Some other updates. Important for the centers is this standardized and centralized Alzheimer's-related dementias neuroimaging, or SCAN. The goal is to provide standardized PET and MRI acquisition prospectively. Any ADRC can upload. You see the sums here for what is currently uploaded. And the last bullet, maybe the most important exhortation, we expect your ADRC, all the ADRCs, to submit and participate to maximize the impact this can have by pooling harmonized and standardized resources across the very important imaging activities that all the centers are engaged in. Next, please. We've had some very productive interactions over past years with veterans, Veterans Administration, over multiple areas, but prominent among them has been those related to AD and ADRD, including the recruitment of veterans into research. An important potential for increasing the diversity of our research. Many of the centers, many of the academic institutions, of course, are linked to VA facilities. And so here through this research initiative, we are funding the infrastructure and collaboration and training that will go into enhanced recruitment of veterans for their benefit, as well as that of the research establishment, into the field of AD and ADRD research. Next, please. This is an ever-growing summary of the active NIA AD and ADRD clinical trials. You can see the total numbers now up to 317, including a very substantial increase in that in dementia care and caregiving, but across the full spectrum of pharmacologic and non-pharmacologic interventions. These summaries are then tied and linked to additional information to allow all of our audiences to appreciate what you can see here. Important emphasis to all of our constituencies, the diversity and increasing diversity in targets and approaches that are prominent at the early stage and now are working their way into the later stage of clinical trials, as we look at the multiple potential targets and pursue all of them and depend on you to continue to generate the exciting and novel ideas to increase and enhance our chances for finding the right target and combination of targets to ultimately approach the problems of Alzheimer's related dementias. Next, please. Want to let you know that after 13 years of service by Marie Bernard as Deputy Director of NIA, and extraordinary service at an institutional and personal level, it's been a delight and a rare privilege to work with Marie. She's left NIA to head the UNITE initiative and those diversity initiatives across all of NIH, another very important task that she will undertake. But it means that we are now recruiting exceptional candidates for the position of Deputy Director. And we encourage all of you, yourself, through contacts, to emphasize the importance of the job. I think Marie is a great personification of the kind of impact the Deputy Director has had and will continue to have in our organization. The search currently is open through the end of November, so a couple of months. And please feel free to encourage people to contact me or the search committee. The link for the details in the search is shown here. It's a very critical and important position for the Institute and therefore for all of us in AD/ADRC research enterprise. Next, please. Finally, these are the links to stay informed. We try to communicate with you in all ways we can to be as near real time and sharing information that's vital to our collaborations. And let me end there and look forward to questions as time allows into the rest of our morning or afternoon session. Thanks so much for the opportunity to be with you.
Thank you, Dr. Hodes. I don't think that we have any questions at this time, so we can move to the next presenter, Dr. Eliezer Masliah from the NIA, is going to give us an update.
All right, thank you, Oscar, and thank you, Richard. Good morning, good afternoon, everybody. Great to be here today, and I'm gonna give you an update of some of the progress that we have made this year or few months with all the extraordinary funding that Richard just mentioned to you. I think we're making great progress both on understanding the relationship of aging, genetics, and environment to Alzheimer's disease, and how this is leading to not only proteomopathy, but also neurodegeneration with all the consequences in terms of clinical outcomes, diagnosis, and therapeutics. In terms of the genetics, we just recently funded for a new round GCAD, NIGAT, and the ADSP follow-up study, which, as you all know, are a very important part of the Alzheimer's disease sequencing program that seeks to have about 100,000 whole genomes by 2025, including diverse populations, which is a very critical role, and in which the Alzheimer's disease research centers are tightly involved. And this program continues to grow, so not only collecting the genomic data and now phenotypic data as well through the Alzheimer's centers and NACC, but also adding a component on functional genomics, which is quite important. I'll describe in a little bit more detail, artificial intelligence, and data harmonization. So all of these together, bringing endophenotypes and measures of polygenic risk scores, really leading to understanding better the heterogeneity of AD-ADRD, and hopefully toward developing a personalized medicine approach. Now, in regards to the ADSP data harmonization program that, as I just said, was just recently funded and put forward, is a U24 mechanism. And what this program is doing is really integrating, curating, documenting, and harmonizing data coming from LONI, from NICRAD, of course, from NACC, with all the phenotypes, images, and genotypes. And all of these is going into a harmonized manner into NIGATs for distribution to you, to the community. And now there is a very recent work group that is also seeking to integrate the work of SCAN that Richard just described to you, and is very well known to many of you, into this process. Now, along the genetics, we also have been doing a lot of progress in molecular mechanisms and processes involved. And we have now a very strong presence in the area of transponsible elements and aging and ADRD, and the RFA just came out, and we're welcoming applications. Also, the Division of Aging Biology along the same lines had one on cytosolic DNA sensing in aging. And we just had approved for our one on non-coding RNA, which I'll be talking more to you maybe the next meeting. But this time I'll talk to you about the functional genomics that we just funded. And this functional genomics consortium has as a main objective, of course, to bring the next level of genomic data and understanding how these late onset genes play a role in Alzheimer's disease, and making all these data available to everybody. I mean, including multi-omic data, cell-specific atlases, epigenetic data, vascular gene data, microglial data, diverse populations. And we funded 6 centers for this purpose, really a wide variety of centers doing different types of functional genomic studies. And again, all of these data going into NIGATs for sharing with the community. And again, the Alzheimer's centers play a major role here. So really here our idea is that in between now the functional genetics, these new transposable elements, and the one that it will come up, the non-coding RNA, we will have a better understanding of the functional genomics atlas for Alzheimer's disease, and really, again, also moving into personalized medicine approaches. Now, talking about aging and aging-related molecular mechanisms, well, here we know that real aging, and because of the genetics of aging, is really a balance between repair and damage. And of course, we have resilience mechanisms and genes involved in these resilience. And there has been some very interesting GWAS studies with over 1 million people showing that, of course, genes like ApoE play a major role, but also there are many other genes that are involved in cell cycle, cell senescence, transposable elements that are quite relevant. And so our idea here was to develop centers for studying those individuals, resilient individuals that are aging well and that we call superagers. And really here the term that we're using is individuals with superior cognitive performance for their chronological age. And we funded 2 centers. One is RADCO, Resilience Resistant to Alzheimer's Disease in Centenarians, with Dr. Perls at BU, over 495 superagers and a lot of controls, a number of biomarker studies, the data going into AMPAD. And the other center, Super Aging, from Dr. Rogalski at NYU, which is a study to uncover pathways to exceptional cognitive resilience in aging. And this one more focusing in behavior, health, genetics, environment, psychosocial. And again, all the data being distributed to centralized AMPAD. And of course, these crosstalk significantly with the Alzheimer's centers, and all the data here I think is going to be very, very useful and important, and the collaboration with the Alzheimer's centers is going to be very important. Just some examples of some of the things that they're being doing in the genetics. Now the recognition of these MAP2K3 loci as a potential loci for in superagers for these resilience, and we hope to see a lot more progress coming up. Now, in terms of molecular mechanisms, also we're putting forward a lot of very exciting funding opportunities. We have had a lot of very exciting workshops, and unfortunately I don't have time to talk about all of them, but I'll just give you a couple of examples. But we have some really exciting stuff going on deciphering glycosylation codes in AD/ADRD. We also have these, and I'll talk a little bit about lipids in brain aging and AD/ADRD. We have the glial cell plasticity. We just got approved for liquid-liquid phase separation that hopefully I'll talk more to you next time. And then I'll talk a little bit more about all the proteomics progress that have been made, and all the proteomics funding opportunities where also there has been absolutely amazing progress, and I'll talk a little bit more about that. So regarding the lipids and brain aging and ADRD, we had this workshop that was organized by the division by Amanda, Paul, and Brad. We had really extraordinary speakers talking about lipid droplets and aging and ADRD, myelin and ApoE homeostasis, and providing really very important recommendations that we took into place when preparing the RFA. As you all know, lipid droplets play a very important role not only in fueling cellular storage, but also now we understand play a role in inflammation, in lysosomal degradation, autophagy. And during aging, I mean, there was data presented showing accumulation of lipid droplets in microglia, probably resulting in deficient phagocytosis, pro-inflammatory cytokine, and oxidative stress. And also the presence of these so-called white matter-associated microglia that clear myelin aggregates, lipid aggregates that form during aging, and that appear to be TREM2 dependent, but not ApoE. And these microglia in the white matter appear to actually precede the disease-associated microglia response that we see in Alzheimer's. So a lot of very exciting, interesting biology. And again, we have that funding opportunity. So for those that are interested. Now, as I said, where there has been also tremendous progress in the last few years is in proteomics. I mean, the technology has grown tremendously, and we have put out a number of funding opportunities in this regard. One on high resolution mapping of biomolecules in brain cells. I mean, I think it's amazing that now proteomics could be done in single cells or single structures, like in this study where they did proteomics in plaques and show colocalization of lipids, including ceramides and phosphatidyls and other things with A-beta. And also these new technology on regulation of the brain proteome in specific cells, where using a combination of specific transgenic mice expressing in selected cells promoters that allow, using click technology, do the proteome in individual cells that have resulted really in very exciting new works. And I mean, we're funding a number of projects in this regard. I told you about this project before, Tom Montin and Mike McCaws on next generation translational proteomics that also is going to be doing a lot of these cell-specific and very detailed high-tech proteomics, and all these data making available via Panorama, and a lot of the data coming from Alzheimer's Center. So again, really very important for you to be aware and take advantage of this. Also very exciting, this just new publication that came up on the new taxonomy for tauopathies. And this is linked to also funding opportunities that we put out on a research network for protein polymorphisms. And a lot of the data on this Nature paper came up from brains that Dino Getty procured through the Alzheimer's Center program. So really very exciting work. And also I want to let you know that now NIA is funding a UniProt that, as you all know, this is the Swiss-based repository for all protein data and protein structure. So we're now funding this center, and you can use this, of course, this center for your work. And very exciting announcement, very recently, the publication from Google Mind on their AlphaFold 2, their algorithm that now they made publicly available, and this site is also publicly available to screen and perform studies of protein folding. I mean, they are describing this as a solution to the protein folding problem, which is really quite extraordinary. I mean, thousands and thousands of proteins for which no X-ray crystallography or data is available. Now computational approaches could be used to understand better that structure. So if we combine all these new technology and proteomics and all the resources that the Alzheimer's centers now have in terms of liquids, brains, materials, etc., I think the future is extraordinarily bright. And just a few words, I'm closing now, on environment. And here, of course, we heard yesterday these extraordinary presentations on CTE. And we are interested also, in addition to TBI, heavy metals, pollution, toxins, and hopefully we'll talk more to you about that in the future. But one area where we've been very interested is infectious etiology of Alzheimer's disease and the role of immunity. We recently funded this RFA on infectious etiology. We got approved for funding one on innate and adaptive immunity. Previously, we found one on HIV and Alzheimer's disease, and we have one active one on neuro-COVID of interest to you. And we just recently had a couple of days ago this workshop, really exciting workshop on infectious etiology of Alzheimer's disease with a plenary by Rudy Tansey, and really very interesting talks on HHV6 and chlamydia, toxo, all kinds of different infectious agents and their role, both A-beta as an antimicrobial agent, but also the role of these pathogens maybe in activating inflammation. Really a lot of very controversial area, but we hope that with these RFA and the programs that we're funding, we'll learn more about it. And again, the Alzheimer's centers here play a major role because most of the data from autopsy samples is coming from you guys. So again, thank you so much to everybody for cooperating with all these people asking for tissues. I mean, your work is absolutely amazing and very important. We also had an NIA Neuro-COVID investigators meeting. We funded a number of supplements in this area. Again, we saw some really interesting work coming from the Alzheimer's Center, from Tom Beach, showing in a small fraction of brains SARS-CoV-2, and also some changes in APP immunostaining. And he has a paper now in Meta Archives on this. But again, I think here we funded a number of supplements to Alzheimer's centers, and we're looking forward to see what you guys find in the brains of these individuals. We actually found, together with NIMH and NINDS, also had a neuro-COVID meeting looking at clinical neurological mechanisms, also at long COVID. And a lot of work was presented as to the effects in the brain on blood vessels of SARS-CoV-2. I think the data on SARS-CoV-2 in neurons or in brain was very little or negligible, but the effects of SARS-CoV-2 in endothelial cells in the blood-brain barrier and damage, activation of microglia, T cells, and other cells, and these vascular, it was quite outstandingly presented at this meeting. And finally, as you all know now, these very large NIH, trans-NIH initiatives for the post-acute sequelae of SARS-CoV-2 or long COVID now has been officially announced by Dr. Collins. As probably many of you have heard, he's retiring toward the end of the year. And this is called the RECOVER Initiative, researching for COVID-2 to enhance recovery. And the centers that are going to be conducting the research have been already announced. And again, all the data on long COVID that is going to be collected will be made publicly available. And there is definitely a very important component of neurological involvement in the studies that are going to be performed. This is more than 15,000 individuals with long COVID. So it's going to be very, very important. And finally, just to remind you, we have had some new initiatives in terms of training. Of course, I didn't talk about training this time around, but I'm hoping to do it in a next opportunity. But we have all these new opportunities for pre-doc, post-doc, and pathway to independency, that is K99, R00, to promote diversity in the workforce and also in the drug discovery area. That is another area that I would like to talk to you in the future. We have these for F31s, F32s, and as I say, K99, R00, really wonderful opportunities for trainees. And again, it's an area that we want to develop more, and hopefully we'll talk more to you in the future. So I want to thank you again and close here, and just remind you to go to our website, You can find all these funding opportunities, the summary of all these meetings, and all these things that I talked to you in our website on these links. Thank you so much. And I believe that next we have Nina with also some really exciting information.
Okay, thank you, Eliezer. As usual, the science that is being generated through the NIA and through the centers is impressive. So next year, we are going to need almost 2 hours for you
Right.
to describe all the things that are going on. I think that we have one quick comment, one quick question coming from Sudha. It's related to the if the BU and Northwestern University, I think you presented in one of the slides, is a minority focus study.
No, this is more like super agers. I mean, there is a small fraction of a diverse population, and they do have a few about that. But we have other efforts going on also in that direction to have even greater diversity in those types of more focused studies.
Okay, somebody clarify that.
Dr. Rogowski answered.
Northwestern University seems to have also minorities.
Yes, that's correct.
Okay, our next speaker is Nina Silverberg from the NIA, and she's the director of the Alzheimer's Disease Research Centers program. Nina?
Thank you. Can everyone see my slides? The right format? Yes?
We can.
Yes.
Thank you. Okay, and you can hear me, I assume. First, I want to thank everyone just for making it this far. I know it has been a really hard 1.5 years for many of us for many different reasons. Some of those are apparent and some of them are less so. And on top of all of that, many of you have been able to volunteer in your communities. And I want to thank you for that too. Doing that on top of everything else that was happening in your lives, in your communities, in your work, with your families. So I also want to warmly suggest that you please be sure to give yourself a break. Everyone's doing more than they possibly can, and it's important to be sure to take a break. We tell people providing care with people with dementia the same, so please take that advice. And I also want to thank all of the research participants and co-participants for their continued interest and giving of their time and helping us move the research forward. I do want to thank again the CTF. I hope it was obvious yesterday to those of you who tuned in. And if you didn't have a chance yesterday, please be sure to watch the video when it becomes available. They have worked so hard to not only streamline, update, and incorporate thoughts from all of the centers, that input that you all provided. There's been a lot, which I think is fantastic. And it was reflected in some of the other sessions. What came out in the other sessions had been incorporated into the feedback and really taken into consideration. So I appreciate the process they've gone through and appreciate everyone providing input. It's so valuable to have people who are actually, conducting the research, giving the feedback, and hopefully feel like it was listened to and incorporated where possible. And really trying to balance what we need to be doing with understanding the burden on participants and staff. I also want to really thank the steering committees and especially thank NACC, because yesterday was just terrific. I think, we had some great talks, great information conveyed, and it's all really important. I want to also say, of course, welcome. Everyone's welcoming the new centers, and it's wonderful. They've expressed to us at NIA their appreciation for all of the welcome feelings that they've been appreciating very much. So I think it's wonderful. We are not only doing great work independently, but I feel that in the past several years, there's been really an effort to leverage the full network and embrace the full network and work together on many of the activities that we had been working independently on. And I think that's also reflected in the work the CTF is doing on the uniform data set. And NACC is right there along with us, helping to make sure that happens. So really appreciate all of the community feeling that has come from all of this work. That said, I do think the Alzheimer's centers are doing quite well in terms of diversity overall as a network. It is certainly going in the right direction, but I don't think that we can rest on our laurels. I happen to have been reading this paper as I was preparing my slides and wanted to share these words of wisdom that are quoted here. Inclusion is achievable when it is prioritized and collaboratively planned. And we see examples of this. I know that people are trying very hard. It means it's important to everyone. But for some of us, it's not our main area of focus. And I think it will help to recognize that there are people with expertise, many of whom are already in our centers and are already working really hard to try to improve our diversity along a number of dimensions. I don't mean just racial and ethnic, but certainly that's important. So AD studies that do not represent the diversity of humans affected by AD should no longer be acceptable. And I will say it never really was acceptable, but it was accepted. And I take responsibility for that as well. Many of us just accepted that it was hard and it took hard work and we tried and we didn't succeed. But it really wasn't acceptable. And I think we need to continue to try, and we need to continue to bring in people with expertise in this area and take their advice and pay them for their expertise. And as such, that the centers are doing well in this regard. We can do better because we contribute so much to the global, the national efforts in Alzheimer's and related dementias. I think it's really our responsibility to continue to improve our diversity. And these contributions also mean that we really are, and I hope to continue to be, leading the way in Alzheimer's and related dementias research. And we have a responsibility to move to novel approaches. And in that vein, I will devote most of the rest of my presentation to talking about how I think we can do that using technology and digital biomarkers. So for those of you who have attended the AD/ADRD summits, there have been over many, many years recommendations about gaps and opportunities that are supported by digital phenotyping, using wearable, mobile, e-consent, and many other innovative measures and approaches for assessment, monitoring, predictive modeling, and other aspects of research. And I think it's important, hopefully you saw Dr. O's presentation yesterday. These are not just to assess the same measures and outcomes we've been using, but really to look at some novel variables that we have not been able to access previously. Some of them may offer an opportunity to have less burden, not more burden, for staff and participants. We may be able to measure things that we were never able to measure before without burdening people. I think this is critical. It's so critical that it had reached in 2016, there was a report, and our own Jason Karlowicz was on this group providing advice and input on this report, Independence, Technology, and Connection in Older Age, that came out in 2016 from the White House PCAST, which is the President's Council on Advisors on Science and Technology. as a follow-up to that, that was recommended that there be a task force put together. And I actually co-chaired this, a report, Emerging Technologies to Support an Aging Population. And so there is a lot in there to focus on what we can learn and what we can do to support older adults to be independent for longer and to understand changes that are happening that may be subtle and that can't be captured easily with an office visit. So I borrowed this slide, with permission from Jason Hassenstab, and Marty Slowinski contributed the figure here, which is meant to convey, as I'm sure you're all very aware, that people have good days and bad days and just variable for performance for all kinds of different reasons. And if you only see them every 6 months or every year, you're only getting a very small snapshot. And if you're only relying on their self-report or their co-participants' report of what changes are happening, you may be missing quite a bit of information. And so the situation that happens when people do come in is that the assessments can be pretty removed from what's really going on. The contexts are not what they're usually in. You all know all these things. I'm not going to list through them, but I think that technology can help us address these concerns. This slide was stolen from Dr. Jeff Kay, who points out some of these same ideas, that when people come in, it's brief, episodic, it's in the clinic, it's subjective, it's obtrusive, and it's inconvenient. And so if we have some of these in-home measures that he's been working on for decades, those are real time, they're continuous, they're home-based, they're objective, they're unobtrusive, and they are ambient. And they actually measure the real thing we're trying to get at in a lot of cases. How often do people take their medications? How well do they sleep? How often do they use the computer? How often do they use the phone? How often do they move around in their house? So I think that this is really a direction we need to be moving. I think that with the presentations at the summit, presentations we've had at the ADRC biomarker meetings from Dr. O, Dr. K, and others, the fact that we put into the RFA for the centers that digital biomarkers are acceptable biomarkers, we're really trying to push in this direction. And the CART program, Collaborative Aging Research Using Technology, was a step in this direction for getting this in-home information. And I think it was a proof of concept because it included people who are in HUD housing. It included multiple different minoritized populations. And some people really like it. Some participants really like it, and it can be made accessible to lots of different groups. So here's the approach that we've taken with the technology work group of the clinical task force that Dr. O is leading. We have surveyed what not only the centers are doing already, many of you are already doing things, but also what the participants have available. And we're working to address some of the perceived barriers and concerns. Dr. O and her team are developing a menu of basic assessments with optional additions. And some of these are already available and some are in process. And the other last piece that's very important to develop this infrastructure to support this work, support the data resources and connection with the other NACC data so that we can do this again as a team, as a network. So the Technology Accessibility Survey has been available from NACC for quite a while now. We have submission of over 2,000 forms and over 2,000 unique participants, but unfortunately only 12 ADRCs have contributed so far. So I'm making sad face to encourage those of you who have not contributed your data. If you have collected it, please put it in. And if you haven't collected it, please begin if you can. But here's what it has shown on those that have already completed it, which I think is exciting and in a positive direction. Over 40% of the respondents are open to video study visits. So that's great. Asking how they currently access the internet. I think, these numbers may surprise people that now most of them do have internet access. Do they use email? 85% said yes. Are they interested in using any of these to have part of their study visit at home? Look at these numbers. It's great. They're open to it. So the survey results from the centers, as opposed to from the participants, all of the ADRCs said they were either interested in or already engaging with digital tools in some way. So this is great. We're already on the right path. Primary barriers that were expressed to using digital tools are participant feasibility and interest. So we've started to address that. It looks like it's possible. Staff burden, access and availability, funding, of course, and validation. So I just, took these off the top of my head, and these are very, very, very legitimate concerns. And we can answer some of them. I'm not going to take the time now because I see we're getting close to the end of our NIA time, but we do have answers to many of these, and others we will need to work on. But I think they should not get in the way of our progress on this very important effort. And I will say that pharma is already pursuing this. I'm sure many of you know this because you're working with some of these industry partners. FDA has issued guidance. FDA encourages the development of novel approaches to the integrated evaluation of subtle early AD pre-dementia functional deficits, impacts that arise from early cognitive impairment. So FDA is on board, pharma's on board. Other countries are doing this, Canada, Europe, Japan, China. They recognize that we need technology because of our growing older population. And this is also from Dr. O. She's working with people in all these other countries already to help with some of their efforts on older adults and technology and assessing cognitive change. So here's what the committee is working on: in clinic, remote, and some cognitive assessments that you hopefully heard about yesterday that can be done either in clinic or remotely. So in clinic, we can, with minimal changes to what we're already doing, digitally measure these existing assessments and record voice. And this can be used, as you also heard yesterday, for really innovative research. And we also can, and some of you are, add new digital measures. Remotely, we can sense gait speed, we can sense room changes, we can add surveys, which is something we've been very slow to do. If we set up this infrastructure, we can ask a few questions, and most people don't mind answering a few questions here and there. So it's not too much of a burden. Some will, some may not have access. I recognize that, and I think it's something we need to pay very much attention to and make sure we're not leaving people out. But I think it can be done, and I think we should be doing it. Last, I just want to cover a couple small things that are not so small, but not related to the previous topic. The best practices guideline update. I've been mentioning this all along, and finally we have been able to complete many of the pieces. And I really thank Dr. Krista Mulder from WashU for organizing all of these different pieces and putting everything together. Many of you have contributed to the individual or multiple of the guidelines, best practices. And so we appreciate all of the effort that has gone into these. There are still a few more, and we have plans to make sure that they stay updated and relevant. So please let us know if you take a look at them and something needs to change. Definitely let us know, but we're glad that they're up there and available for review. And last, please mark your calendars. We have planned a biomarker meeting. Dr. Elliott is leading that. And it will be on Wednesday, November 3rd from 11 to 3 Eastern. And the purpose will be to gather the current state of the biomarker cores and discuss future plans as NACC prepares to establish a biomarker core steering committee in January. And the agenda should be coming out next week. And then last but not least, Dr. Nelson from Kentucky and Dr. Schneider from Rush are organizing the late update meeting to provide new and/or interesting data on TDP-43 pathology in aging. And they are very interested in receiving any work that you have on this that you would like to present as a poster. So I think that was my last slide.
Okay, let me see if we may have a question. We don't have a question, I think, Nina. So very, very impressive. I mean, all the science that has been generated through the ADRCs. So next year, you and Eliezer will have almost 3 hours to present. Our next speaker is Dr. Kukull from NACC, and he's going to give us an update.
Okay, here comes my slides. Good morning or afternoon, as the case may be, to everybody. I'm happy to be here virtually. I wish we were in person. I'll try to be pretty brief today. I think the theme that I'm getting at here today is really the resilience of the centers. So let's look at the first slide here. There's been some changes. NACC has been refunded now as a U24 through June of 2026. Back there we go. And there's been a lot of changes at NACC internally as well as in other sorts of things. So Carrie Stevens, who was one of the associate directors, along with Sean Mooney, Gary Chan, and Charles Mock, is now the deputy director. We're welcoming also new people in terms of program director Sarah Biber, our new data group technology lead Ben Keller, and Valerie Hawkins, a grant management specialist, who is actually taking over the duties for Rado Lee, who will be leaving for another job here very soon. So, okay, I wanted to go on here to the UDS4, and if you look at that comment there that I made, you're going to love it even more than UDS3. It kind of goes what every time I say UDS4, people around me usually have a heavy sigh, and it's kind of like it's a little bit foreboding and it's a little bit optimistic, but I think you should consider it much more optimistic than that. The clinical task force has really dug into this and tried to keep it up with the best science, and also keep an eye on what actually takes place on the ground in the centers as to how these things are implemented. So it's really kind of an amazing thing, and I think we should try to eliminate that heavy sigh that goes with every time you say UDS4. It shouldn't be, it's really much more optimistic than that, and they've done really tremendous work. And I hope you'll all go through these draft forms as they come out to your center and try to help the clinical task force in finalizing and adding comments that you may consider relevant, that maybe somebody hasn't thought of yet. So anyway, I think that's one part of this sort of aspect. And another part of it is, at NACC, we're trying to modernize our systems to keep up with all these changes. we're implementing REDCap in a wider way, and also this new data visualization cohort formation tool called LEAF. You'll hear much more about these and also the personnel changes at NACC at the session later today. So I hope you'll all tune into that. So this whole thing, all these changes that are going on, it's really about the resilience of all of us to a lot of different things. Resilience of NACC to changes in personnel and to expansion and so on. Resilience of the centers to UDS4, how we attack all those sorts of things. And can I have the next slide, please? So I also wanted to comment about the resilience of the centers and really compliment the centers. Over the last year or so, we've really had a lot of adversity from the standpoint of COVID and how the centers would really work to counter all that with the increasing challenges. So as you can see here, that in 2019 there was about 8,500 follow-up visits, and in 2020 there were 6,500, and that's when you look at the composition of those being in 2019, about 1,000 of those were done remotely, but in 2020, about 4,200, or a much larger chunk, were done remotely. This was all due to the rapidity that the clinical task force developed remote UDS and the TCOG, the neuropsychological battery, and the COVID survey that Nina mentioned, that we could have maybe a little bit better participation on the COVID survey. But the resilience of the centers to adapt to this COVID challenge by using a whole completely different way of doing things to start collecting remote data is pretty remarkable, with all of the changes due to COVID. So I wanted to really compliment the centers and the clinical task force for pulling all this together so rapidly and for responding to these things so quickly and really completely, because the number of visits is probably that decreased in 2020 was probably a lot due to COVID itself and not anything that was going on at the centers particularly, but it's really particularly remarkable that the total number of follow-up visits is not much changed really from ordinary practice. The next slide, please. As with some of the other things I mentioned, or the heavy sigh after saying UDS4, this slide also is really not what it seems as being a Halloween slide. This is more of a music trivia slide that maybe many of you understand already from looking at the picture, having to do with Edgar Winter. But I think one thing you can take away from here in terms of the previous slides, and I was mentioning the resilience of the centers, the clinical task force, and really everyone in dealing with the adversity that we faced through COVID, and all the participants at all the centers have faced as well. So, because this really is a music trivia slide rather than a Halloween slide, I think you should always remember that you're never too old to rock and roll. Okay, that's all I have.
Thank you, Bud. I missed one comment. Can Kathleen Welsh-Bonner said it would be great if digital technology we use across centers follows strict guidance to ensure the CFR Part 11 compliant. And Jennifer Lingler said, but can you touch on how the pandemic impacted recruitment and retention of persons with underrepresented groups?
I don't know if I can comment too much. We, the clinical task force is certainly debating all these things, and you saw the presentation by Dr. O yesterday about all the kinds of digital possibilities that we have out there. Those haven't been implemented at this point. There's a lot of pilots potentially planned that the clinical task force is working on, and so I'm sure a lot of the considerations with such digital measures will be addressed along the way. We're not quite down that road yet.
Okay, thank you, Bud. So we are going to move to the scientific sessions. Our next speaker is Angela Taylor, and she will talk about the Nomenclature Initiative.
All right, one moment and I'll get the screen sharing started. All right, are you able to see my screen?
Yes, we can.
Okay, wonderful. Well, on behalf of the steering committee, I'm really delighted to be here to share an update from the Dementia Nomenclature Initiative. So we're really grateful for the opportunity to provide you an update. My job today is to provide you with a preview of our work, and I'm pleased to have Ron, Sandy, and Jason here, as Oscar said earlier, to add to the discussion and to take your questions. I'll share just a few examples of some of the communication challenges we're looking for solutions to, and also a little bit about the origin of the initiative. But mostly, I'm going to focus on our approach, and I'll end with the communications framework that we've developed. One of the reasons we're here today is to hear your questions and comments about what we're proposing. So there will be a 10-minute time slot at the end for discussion, and I really encourage you to submit your questions to the Q&A. If you have comments or want to discuss this, please do put those in the chat box. All right, so what's the issue? Clear, accessible communication is at the heart of promoting public health. But right now, the public is struggling to understand what are very, very simple concepts. What's the difference between dementia and Alzheimer's disease? Is dementia a mental health issue or even a personality defect? Is it just a part of aging? And this confusion can actually create serious harm. And they're not the only ones impacted by the terms that can be misinterpreted. Researchers, clinicians, and advocates don't all communicate in a common and consistent way. Collectively, these challenges can drive stigma and isolation, delays in diagnosis and care, and barriers to more effective public education efforts. And it complicates matters when it comes to policy and research as well. So our goal is to identify opportunities to create more clarity, more consistency in how we talk about these conditions, so that together we can all drive increased awareness and public access to care and research and reduce the experience of stigma and advance scientific knowledge, policy, and legislation for the public good. Early interest in this initiative grew out of a hot topic session at the 2016 Alzheimer's Disease-Related Dementia Summit and built momentum over the next 3 years through presentations and recommendations at 2 subsequent summits and echoing recommendations which were issued by the Federal Advisory Council on Alzheimer's Research, Care, and Services. The Dementia Nomenclature Initiative was formed shortly after the 2019 summit to implement the recommended transparent and inclusive process to explore opportunities to improve communication around dementia and the diseases that cause it. Over the past 2 years, we've had a lot of discussion about what our scope of work should and should not be. Our scope is limited to the diseases listed in the NAPA legislation: AD, FTD, LBD, VCID, and mixed dementias. But we also want to reassure you of some things that we are not doing. They are outside of the scope of our work. We are not revising any criteria or definition of any disease. We're going to leave that to the scientific community. We're not recommending replacement terms for the labels that you're currently using, which may seem like the Nomenclature Initiative is a misnomer. But what we are doing is we are going to offer you a new approach that we can all use consistently, that will explain this full spectrum of cognitive impairment and the diseases that cause it. And our work is not meant to influence any funding priorities. The communications framework we've created is agnostic to any single disease. So, as Oscar mentioned, we have 3 working groups that have taken on different aspects of this issue. One focused on research, one for clinical care, and one for public stakeholders. The early efforts of the research working group centered around deconstructing Alzheimer's disease and related dementias, which all have variations in their clinical presentation, syndromes that have distinct and also overlapping clinical features with other dementia syndromes, and commonly mixed pathophysiologies. We started by identifying the main common elements, and then we began to break those down a little bit further. So, for example, one element is clinical presentation. What are the symptoms? How severe are they? And how much do they impact function? And for research purposes, at what ages did they begin? This adds up to the element of the clinical syndrome. Another main element is the disease process or processes that may be at play and what evidence of them may be available clinically through genetics, biomarkers, and later at autopsy. Lastly, we envisioned one additional element to minimize public confusion and stigma in the form of a single overarching term for these diseases. One of the key considerations for the framework was that it should be representative of the full continuum of the progression of these diseases from the preclinical stage all the way through advanced dementia. And so that's been an overriding factor in how we built the framework. The other one is our desire to have this be interoperable, that this is a resource that works for multiple audiences, not just the scientific community. So how do we take those very complex concepts and then break them down in a way that we can more readily explain them to a person who's concerned about their memory or their function? Well, we decided to start laying them all out in a visual construct, and we used terms that could be more easily understood by everyone. So knowing that there could be broad implications, well, not could, there will be broad implications for any changes that are proposed for nomenclature. We also made it a priority to solicit some preliminary feedback on the framework, both from members of the public who've been affected by dementia and also from professionals in other key sectors. We started with a half-day workshop for NIH stakeholders on nomenclature challenges in research, and then we presented a preliminary version of the framework for discussion. We engaged SCP, a communications firm with experience in aging issues, and they ran 5 focus groups of caregivers and people with dementia from different ethnic and racial groups, so that we could better understand how today's terms are or are not meeting their needs, and to seek some early feedback on whether elements of the framework would be helpful to them. And we also had RAND run some focus groups to seek early feedback on potential opportunities and gaps in the framework from professionals from key sectors who are not engaged in this initiative yet. And we were looking for early red flags and signs of whether or not we were moving in the right direction. So here you can see how we laid out the main elements I mentioned, again, with some additional details for each of them. At the top is the umbrella term, and we're working on an umbrella term that will function conceptually like the term cancer, one that's scientifically accurate, that's appropriate for multiple diseases that begin silently and later present clinically in different ways. And I'll share more on our thinking on the umbrella term in just a minute. Moving down next in the framework is clinical presentation, which is divided into the various features, and we've provided qualitative severity ranges. The features block itself here on the left is not meant to be all-inclusive, but it is inclusive enough to allow for multiple dementia types to be represented in it. In the middle column, you see we have severity ranges. One of the things that you don't see is the term mild cognitive impairment or the term dementia. We've broken these into functional severity ranges and then domain-specific ranges. We've used qualitative terms that clinicians can use to help their patients and families understand where they are on the full trajectory of change. This is something that we heard very clearly from the public stakeholder working group and the focus groups of caregivers and people with dementia that was really needed for their own future planning purposes. For research purposes, we did align these with the current terms. So minimal is parallel to MCI, and mild, moderate, and severe reflect the range of dementia. And you'll see here that we separated the clinical syndrome from the underlying disease processes. And we felt this is critically important, that all stakeholders understand that a variety of diseases can drive cognitive change and that these diseases require a differential diagnosis and treatment. The umbrella term, excuse me, I feel like I have a hair on my mouth. The umbrella term is still under construction. So what I want to focus on is why we feel one is needed and the process that we've used to get to a working umbrella term. The term dementia was established in the 13th century, about 700 years before we came to understand its many causes. And in the 21st century, that term and the term Alzheimer's disease are often now used synonymously, which is not accurate. And we also see terms disease paired with the syndromic dementia in what we currently use is the closest thing to an umbrella term of Alzheimer's disease and related dementias. So we have a disease term in there and a syndromic term, which might be adding to some confusion. Knowing now what the most common causes of dementia are, with the advent of biomarkers and disease-modifying treatments, we're seeing an opening up of the need to have terms that are reflective of where the field is going, and that's the preclinical and prodromal phases. So we need a term that embraces that full continuum of disease progression, not just the late clinical stage of dementia. So we charged each working group to independently develop their own ideas for umbrella terms. And while they were doing that, we also created a criteria for what we thought would make an umbrella term most useful. First, it should be disease agnostic, but also stage agnostic. As I said, we wanted to represent the continuum of disease, not just the symptomatic phase. It should be specific enough that the name suggests that cognition is impacted. It has to be reasonably lay-friendly and have the potential to mitigate stigma. And lastly, it has to be accurate enough to serve the research and clinical care communities. So with this criteria in hand, all of the working groups then narrowed their suggestions and we found that they had terms that overlapped considerably with this narrow list. So we took all of those terms, sifted through them, and we now have created a working umbrella term that meets the needs of these requirements and has been found acceptable by all three working groups. That umbrella term, which is a working term right now, so don't get too excited, is adult cognitive diseases. And we're still wrestling with a syndromic alternative because we recognize that that's what everybody is currently used to of adult cognitive disorders. So we still have work to do, as you can see, before we settle on any final term, but we wanted to share the approach with you so that you had a sense of where we were coming from when we do come out with any recommendations. So the creation of the communication framework, the working umbrella term, and more than a dozen written deliverables represents more than 2 years of discussions and the help of about 50 collaborators who you see here on screen that range from academia, federal agencies, healthcare and service providers, and advocates. We're going to be wrapping up this exploratory phase of identifying opportunities to improve communication about these syndromes and diseases sometime in 2022. And we're already starting to explore what a next phase of this initiative could entail, which would bring in a much broader array of stakeholder input. So with that, I want to acknowledge our very generous sponsors, without whose support we absolutely could not do it. But as you can see here, they all came to the table. We have this all-in approach on this complex issue, and many of these organizations have somebody that's participating in one of the working groups. So I'm going to stop there, and I'd like to start off the Q&A session with a question both to Ron and Sandy and Jason. I'm going to move our slides back to the framework, and I'm going to leave that on screen in case that's helpful.
Yeah, thank you, Angela. So we can start with a question from Suzanne Kraft. Do the cognitive change categories require that the condition will be progressive?
So Ron, would you like to take that one?
Sure. Thanks, Suzanne. We've discussed that, and yes, it's most likely that these conditions will be progressive. One could think of a vascular situation where it may not progress rapidly, but in general, that's true. And one of the other features that the progression question touches on is something we heard from virtually all the focus groups, and that is, what does this mean for me? If you characterize me as having a disorder thus and so, what does that mean for me in the future? So the prediction, the prognostication element of that is probably beyond what we're talking about right now, but clearly has to be considered. But in general, yes, these are progressive disorders.
Thank you, Ron. Next question is from Andrea Bosoki. Why not adopt the term utilized by neuropsychologists and already in the CPT lexicon, which is minor and major neurocognitive disorder? You could add minimal neurocognitive disorder if you want to expand the spectrum.
Angela, maybe I'll take that one. Yeah, I'll say a couple words about that, because I think what you're referring to is the terminology that came out of DSM-5. And DSM-5 dealt with many of these same issues, the terminology, how do we characterize this diseases, and came to the conclusion that we should describe the disorders syndromically and then get into an etiologic or a pathophysiologic explanation. So very similar to what's being proposed here. The term, now DSM-5 only dealt with individuals who were impaired to some degree. So the term mild neurocognitive disorder was adopted to sort of parallel MCI. Major neurocognitive disorder was meant to parallel dementia. However, as we've gotten into this exercise, neurocognitive disorder is not real popular among the lay public and maybe not among the professional public as well. So that's why just cognitive diseases or cognitive disorders were thought to replace neurocognitive. But the thinking that goes into what ended up being DSM-5 and what's being discussed here are quite similar, actually.
Thank you, Ron. The next question is coming from Charlie DeCarly. Other than change in name, what is new about this approach? We always separate syndrome from etiology and focus on all the clinical features.
This is Sandy, I think. Can you hear me?
Yes.
Okay. I can't see myself, but that's okay. I'll take this one. So yes, absolutely. We always separate clinical syndrome from etiology. However, that's not really true in the general public. And also when you get a research paper, for example, that says we had 300 people with Alzheimer's disease participate in this study. The term Alzheimer's disease is used indiscriminately to refer to Alzheimer's dementia, Alzheimer with biomarkers, Alzheimer without biomarkers, Alzheimer's with other things. So I think that the approach is that we want to try to bring this level of complexity to a point where everybody can understand it and use similar terminology at the clinical level, at the public stakeholder level, and in research.
And I will go ahead and tack on to that with a perspective of, we also have to make those names distinct so that they are easy to remember and differentiate by the public. And I offer, my favorite example is the difference between Lewy body disease, Lewy body dementias, and Lewy body disorders. That's far too difficult for the public to be able to understand. And so I think we have opportunities to be more distinct over time, but that will take time and discussion.
Thank you, Angela. And Mary Sano asked about have you considered that adult cognitive disorders is a common complaint in a wide range of psychiatric disorders like post-traumatic stress disorder, schizophrenia, etc.
Ron, do you want to take that one?
Sure. Yes. In fact, the pushback that we've received about adult cognitive diseases or disorders is, where do you draw the line? Now, remember what Angela presented first at the beginning of her talk was that our charge came from the NAPA Advisory Council, and the NAPA law referred to Alzheimer's disease, Lewy body disorders, frontotemporal degeneration, and vascular cognitive impairment dementia. So just those four. But we appreciate if you throw a term out like that out to the general public. What about traumatic brain injury? What about Parkinson's disease, dementia? What about ALS with cognitive impairment, et cetera? You're really bringing in a lot of disorders. So we've tried to constrain ourselves to what our charge was, but this is one of the concerns we have about the umbrella term as to how people are going to appreciate it.
Yeah.
Thank you,
I
Ron.
would also add on to that, Oscar, that one of the things that's really important is that this term requires a definition. And this term is really to represent diseases where the primary driving symptom will be cognitive impairment that's disabling. And I think that's the distinction there, is that these other diseases and disorders may not have that as the central feature.
Okay, thank you. This is an interesting question from Alison Lindauer. Have you considered having a similar nomenclature examination of the term caregiver?
Who wants to take that one?
This is an interesting question.
I'll go ahead and say that there are a lot of things that we could talk about where the terminology is not as successful in all areas as it could be. The term intervention is a good example. The term caregiver versus care partner versus care provider. There are a lot of examples like that, but to our scope, our focus, and the amount of time it takes to get to this kind of stage is extensive. So that might be something another group of professionals might want to take on.
I can just add that in Europe, they use the term carer rather than caregiver. So it does have certain implications.
And Mary Sano said, shouldn't it include change in cognition in age and not just deficit?
Yeah, you're absolutely right, Mary. And it's this kind of feedback that's going to be really helpful to us. We really appreciate all the comments and the, what might seem understandable to us because we've been meeting for 2 years. It's very good to get the feedback on what's not getting across, or things that we haven't considered and things that we need to consider.
Yeah, Eliezer brought this up when we presented this to NIH leadership. And what about cognitive changes of aging? What about subjective cognitive impairment and the like? So these are all relevant topics for us to consider.
I'm not sure how we're doing with time, Hannah. Can you tell us if we are okay with the time?
You have more time.
Yeah, you
Okay,
have a great amount of times. Yeah.
Ray Bateman said, as an etiology, good strategic infant dementia or TBI related cognitive dysfunction be considered as an ACD. They are static and may not be progressive. I can see this becoming confusing if small vessel disease equals ACD and strategic infant dementia is something else.
I think that this comment just highlights the plethora of terminology that we have that is in our field, and that, what you label as strategic infarct dementia could be translated into a static encephalopathy of multiple domains. So we're trying to make it more understandable at the level of the patient who's being affected by these disorders. I think we decided to, as Ron pointed out, focus on the NAPA charge of, just the diseases that we have listed under possible disease processes.
Okay, thank you, Sandy. And questions keep coming. John Olichny from Davis. I like the schema, but question the messaging that MCI has minimal cognitive change and suggests very mild instead. In the clinic, I wish to heighten awareness of dementia risk and not minimize it. For umbrella, brain degeneration may be a candidate, but doesn't include vascular cognitive impairment. Hard enough for us specialists to separate from neurodegenerative diseases.
Yes, John, you raise many relevant issues that we've been trying to wrestle with. Are these progressive? Are these just degenerative? What about the vascular components? And at one of our previous workshops, Julie Schneider, of course, did a very nice job showing us that most often there are multiple processes operating in any given brain, especially in somebody in their 70s or 80s who has some degree of cognitive impairment. The rule is that there are multiple processes operating. So coming up with a terminology that will capture that on the one hand and be acceptable to all the different constituencies and understandable to the lay public is the major challenge. So I think the approach that we took was to try to deconstruct all of the different syndromes, all of the different conditions, what goes into them, how do we make the diagnosis? Do we in fact deal with it purely syndromically? Do we deal with it purely pathophysiologically? Or most often, most of these four disorders really are a combination of clinical presentation and underlying perhaps biomarker profile to the extent they exist. So we have to take all this into account. And I think just bringing this awareness to us as researchers, to the clinicians, and to the public, because this is how we arrived at these diagnoses, will be illuminating, we hope.
Thank you, Ron. Ali Atri asked, what was the consideration regarding not including behavior in the umbrella term? Adult cognitive behavioral disorders, for example, given that we want to include earlier and broader recognition, behavioral changes are often noticed earlier than cognition by family patients, etc.
I'll go ahead and address that. So that was not a popular term among the working groups. And I really think that stigma plays a role in that. If you label this as a behavioral disease or disorder, I think you're going to see pushback because behavior is a result. The behavior change is a result of the disease. Yes, it might manifest earlier, but what would the implications be from a stigma standpoint? And that's one of the issues we're really trying to address here.
Okay, next question is from Jennifer Linkler, but I think this question is for Pat Kukull that we can address later. Gil Rabinovich asked, how does this framework deal with patterns of clinical features that often converge into a well-recognized neurodegenerative phenotype? In some instances, these phenotypes can predict the underlying disease process with high accuracy, even in lieu of biomarkers, mutations, or autopsy. For example, PSP, the progressive, the semantic variant PPA, etc. It may mean more to patients to label their disease as PSP than motor executive mood disorder.
I would like to take a try at this one. You're absolutely right, Gil, that this is the problem. We have the clinical symptoms, which is what people live with daily, and then we have the pathology, which, as we know, is related to clinical profile in a very probabilistic manner. So it's likely that if you have a prominent aphasia and you have comprehension deficits and it's progressive, that you can label it whatever you want, but the truth is that most of the time you'll find TDP-43, but sometimes you may not. And the same thing is true with PSP. You can have PSP syndrome, and we all know that most of the time we have PSP pathology, but sometimes we don't. And so that, we're dealing with this all the time when we have responses to our families from autopsy reports that they get. You said the patient had semantic variant PPA, but they have Alzheimer's disease. What does that mean? So it's trying to get to a level of really helping people understand this discrepancy.
And I might just add, Gil, I don't think we're proposing replacing those terms with the terminology you see in this framework. Rather, we're trying to help the clinician, the researcher, the patient, the family understand those syndromic terms, PSP, behavioral variant FTD, Alzheimer's disease, in terms of its components, and hopefully that most of the components are captured in this framework. So you would still use PSP as the diagnosis, and you would just say, gee, this particular clinical profile is highly suggestive of this condition. And to the extent we have biomarkers that confirm or don't confirm, we can add that to the picture. So again, we're not proposing replacing any of those terms.
Thank you, Ron. And I think that we are going to take the last question from Alison Rosen. Do you feel the public understands the term cognitive, regardless of education or acculturation? Honestly, having multi-word terms are difficult to manage in publications.
All right, how about if I take that one?
Okay.
This we discussed extensively. As a layperson, I take the stance that the public is able to be educated. They're able to take terms that are not currently in the lexicon that they use every day and learn them. The acronym combined with the umbrella term would ultimately allow the public then to shift to ACD. What we are mindful of is the ability to translate, which is difficult with terms like dementia in Chinese and in Spanish because of stigma. I remember as a child learning about multiple sclerosis and muscular dystrophy. Those terms they're not considered lay friendly, but we do learn them. And so we do think this is going to be an educational effort to add that term into the household word status, but I think that it's very achievable. We also, like I said, we intentionally were looking for something that would reduce stigma. And when we go back to the criteria for the umbrella term, and there's a couple of comments in here in the chat box that ask about disease versus disorders and whatnot. When we look at this term broadly across all of the criteria, this was the one term, granted it's 3 words, but you can reduce it to the acronym, that everybody felt was acceptable and met these needs. The ability to move towards personalized medicine requires that we are able to identify these diseases before they're symptomatic. The ability to make this term automatically resonate and not people think, well, if you call this neurocognitive, I mean, that's even harder for the public to understand. So we really do recognize that there's going to be an educational issue, but we think that it's something that is achievable. So I'll just stop there.
Thank you, Angela. Unfortunately, we cannot answer the many questions that they keep coming. So we are going to move to the next presentation.
Actually, we have a break scheduled at 8:55. So we could take more questions if you want to, or we could save this cushion time for Q&A for future
I will finish. We have, I think that we can do a couple more. Rajan Dwara asked, the problem with adult cognitive disorders for the public is that it's not limited to a single word. However, the terms ADD or OCD are now in common usage to the lay public. So adding ACD in parentheses to adult cognitive disorders may help common usage.
We agree. Thank you.
Yeah.
Thank you.
And Ellen Wixman, I agree with Mary and Allison, the public should be educated on cognitive with modifiers. For example, gradually progressive, intermittently progressive, stable, reversible, early adult onset, late adult onset. The cognitive term can be specified as neurocognitive or psychocognitive. Disorder is better than disease. DSM has dealt extensively with this issue. Minimal, minor, and major can be used to be consistent with DSM-5.
All good comments, and I think that's right. I think this framework does dovetail quite nicely with DSM-5 for the most part.
Okay, I think that we can save time for more questions at the end of the scientific session. I've been following the questions and answers, but there's also significant activity in the chat. So people can continue talking in the chat. Thank you, Ali.
Yes, thank you all for your grand participation today in this discussion. It's been really rich. So we are going to take a 10-minute break then. We will come back just shortly before the top of the hour.
So Hannah, are you seeing that okay? If I go to the full screen mode, does that look reasonable to you?
Yep, that looks like it's just the slide, no notes or anything, so looks great.
Yeah, that sounds good.
All righty. Well, Oscar, do you have an introduction of Paul?
Yes, I would like to introduce Dr. Paul Thompson. I think that everybody knows Dr. Thompson for the quality of research that he does, and he's going to talk about the ENIGMA. Paul?
Okay, thanks so much, Oscar, and morning, everyone. I really appreciate the invitation, and it's a real honor to speak with you today. So I'm going to tell you a little bit about two very large-scale studies. One is ENIGMA, a global brain imaging study, and the other is AI for AD, which I'll mention at the very end, which is a similar idea for multi-cohort analysis of data sets on Alzheimer's disease. The overall goal of ENIGMA is to look at how now over 30 major brain diseases affect the brain across 45 countries, and to discover factors that affect patient outcomes. And so this has led to the largest neuroimaging studies to date of 13 major brain disorders, and also efforts to screen the genome for genetic loci that affect the brain. One paper I'll mention in Science from last year includes data from 60 cohorts. Many of them ADRCs and center directors were really great contributors to that. And I'll mention at the very end a little bit on AI for AD and these ideas being adapted to machine learning and AI for multi-cohort data. So many of us are familiar with collecting very large databases of MRI, PET data, in some cases neuropath data, and also whole genome sequencing data and other omics data, with an interest in understanding the relationship between each of these biomarkers and how they predict patient outcomes. And so since 2009, ENIGMA began to publish the largest neuroimaging studies of a number of brain disorders, analyzing brain scans and genetic data from now over 100,000 people in 45 countries. And this has led to the largest brain imaging studies of Parkinson's disease, epilepsy, bipolar disorder, major depression, a number of psychiatric conditions, and also neurodevelopmental conditions, OCD, ADHD, and autism spectrum disorder. So there's two main goals of ENIGMA. One is to understand the effects on the brain of these disorders through MRI, diffusion imaging, resting state, fMRI, EEG, and MEG. And then a more challenging problem of understanding how variations in our genetic code are associated with the biomarkers that we care about, to discover factors in the genome and environment also that affect aging and also development, disease risk, and treatment response. So this is where the data is. The colors are the diseases that each center studies. These are MRI data sets that are paired with genome-wide genotyping. And I'll tell you a little bit about how this data is analyzed. So there's been some summary papers talking about the harmonization efforts and different aspects and considerations in merging this data. It's all organized into working groups. So there are now over 30 clinical working groups that focus on different neurological and psychiatric and neurodevelopmental conditions. And obviously the clinical questions for each group are a little different. And also there's a lot of heterogeneity. So if a working group focuses, for example, on TBI, that can be in the military, it could be sports-related concussion, or it could be brain injury in childhood. And obviously the clinical and outcome considerations for these disorders differs a great deal. Paired with that, there are data harmonization groups. So if you're analyzing data from DTI or resting state coming from different scanners and protocols, there are standardized pipelines that we and others have developed to process this data in a harmonized way. And similarly for GWAS and CNVs and epigenetic and omics data, there are standardized pipelines that can be applied to different cohorts. So this is just an overall picture. ENIGMA is studying neurological and psychiatric conditions, a number of developmental conditions, with the techniques that are summarized at the top, different approaches to analyze neuroimaging and other biomarker data. And here are some of the folks leading some of these efforts. And just to sort of tell you how this consortium is organized, it's much like ADSP and many others, where there are groups studying different aspects of a disorder with different methods. And then across disorders or across disease subtypes, these lines here are collaborations among the participants in the network. So this led to the largest studies or most well-powered studies of a number of conditions. These are maps of gray matter, cortical thickness reductions in major depression, bipolar disorder, schizophrenia, also in alcohol use disorder. This will come as no surprise to many of you. I mean, many of these conditions are very heterogeneous and complex in their effects here just on cortical thickness. And we're very interested, obviously, in not just discovering the main effect of disease, but also modulators in the genome or in treatment. And you can also get a t-shirt with this data on. So let's look at the data. So in ENIGMA's lifespan project, brain measures are derived from cohorts across the world. This is just the size of the thalamus in the right hemisphere, and it's an age plot that is a composite of many, many different data sets across the lifespan. If you look at it carefully, and this is, again, no surprise to those of you that are running ADRCs, each cohort is contributing information from different parts of the lifespan. This is left hippocampal volume. In every scan, the dot is a measure of left hippocampal volume. And the colors are different cohorts. So they're different, they may be ADRCs, they may be neurodevelopmental or public data sets. And you can begin to chart the lifespan trajectory, again, based on cross-sectional data. For the morphometry here, the volume of the thalamus and the amygdala, the hippocampus, as you know, declines in volume steadily and then more precipitously later in life. So you can begin to have normative charts that will then reference incoming data to these statistical norms. And you can ask questions about how could you stratify this by sex, by ApoE, by other conditions that might influence the morphometry and the trajectory. So looking to genetics, you can imagine combing the DNA, much as in a GWAS, for markers that influence the trajectories of brain aging. And many of you are pioneers in this. I don't need to explain it. The premise is that genetic variants might affect brain biomarkers or biomarkers in general with greater precision and statistical power than if we were looking at clinical measures. But that's a hypothesis. It may not be true, and we'll look at it. So one cautionary note came from Nick Martin, who was the president of the Behavioral Genetics Association. And he said when we began this, if you want to discover genes or more precisely genetic variants that affect the brain, you'll need brain images from 50,000 people. Just because images are more expensive to collect than measures of height or weight, it doesn't change the power calculation. So we'll see if Nick was correct. He was actually correct. So this is a study in Nature from a few years ago. Many of you contributed to this partnership between ENIGMA and the CHARGE Consortium, in which Sudha Seshadri, Charlie DeCarli, Claudia Satizabal, Oscar Lopez, many others contributed. The question was, where are the common variants in the genome that are associated with the volume of different subcortical structures? And just to explain how this is done, much as in any genome-wide association study, you march along the genome in imputed data and ask the question at each locus if the common variation at that locus is associated with the volume. It could be the size of the brain, or it could be some other measure, amyloid or tau deposition, or something derived from the images. So much like in the AD GWAS, this is Brian Kunkel's recent GWAS for loci associated with Alzheimer's disease. Many of these loci come up as case control differentiators, and we're interested in whether or not they impact brain measures. Some of them may act through other pathways, and some may act through imaging. So ENIGMA members compute brain measures from their scans. With MRI, it might be free surfer. With diffusion imaging, it might be TBSS. There's also protocols for EEG and MEG, and more recently resting state. And initially, they test associations between these measures across the cohorts. And over 1 million SNPs, common variants in the genome. There's harmonized protocols for genetic imputation and QC. And then we meta-analyze the data. So the vote would obviously depend on the sample size. So if you had a cohort of 100 patients, for example, and let's say the UK Biobank has 46,000 individuals, each cohort would get a weighting that depends on the cohort size. And this was initially very successful. 6 genetic loci were discovered that are associated with hippocampal volume. Because these are brain images, you can then do statistical parametric mapping to see exactly where it is in the image. This is the anterior hippocampus that here shows atrophy or volume variation associated with variants in the harakiri or HRK gene. It's interesting what this gene does. There was a study actually of an ethnic isolate in Dagestan in the Caucasus Mountains by a Russian geneticist, Kazima Bulayeva. And she found that the same locus where common variation affects hippocampal volume, when it is deleted, it's associated with mental retardation in genetic isolate. So this is very, very interesting data on what these hippocampal variants might be doing. This partnership between ENIGMA and Charge grew. It became a 30,000 MRI GWAS, and now the 6 loci associated with hippocampal volume initially 5 years ago are now 51. And you could ask, do these genes operate all the time? Do they just operate later in life? And it's certainly the case for ApoE, as you know, that the shift in hippocampal volume in carriers of ApoE4, the late onset AD-associated haplotype, is emerging throughout life. So in early adulthood, there's no detectable difference on MRI for hippocampal volume in the 20s and 30s, but there's a steady increase. Each dot on the plot here is a cohort, and 60 cohorts data have been aggregated to show that by the age of 70 or 80, there's a 2 standard deviation shift in hippocampal volume in E4 carriers versus E3 carriers. And so you could look at all of these loci that affect hippocampal volume and brain volume overall. And you'll notice links with other genome-wide screens of Parkinson's disease and Alzheimer's disease, where certain loci come up as influencing the brain and disease risk. So this is a screen of intracranial volume, a measure of overall brain volume. The top hit is in MAPT, a very important degenerative disease risk gene. And you'll remember in Andy Singleton's GWAS, Andy will speak next after me, for Parkinson's disease, both MAPT variants and synuclein alpha variants are among the top 2 Parkinson's disease associated genes. So you can see this picture is being built where some of these disease risk genes might operate through signals we can detect on imaging, and others, of course, do not. I mean, there are molecular cascades that they influence. So this work by Claudia Satizabal, who I think is on the call, and Sudha Seshadri and Charlie DiCarli with Charge, partnering up with us in ENIGMA, revealed that the genetic architecture of loci that influence each subcortical volume is largely distinct. And so this is very interesting information, that the architecture of variants that influence the volumes of different subcortical structures differs, which implies that there is a genetic mosaic in the brain that affects in a modular way the structures that we care about. Now there are over 51 loci in the genome that are associated with hippocampal volume. And one of the interesting things is, I mean, you could say, well, do these SNPs explain much? They do. So a polygenic score, prorated from the top hits in the GWAS, accounts for just under 5% of the variance in hippocampal volume. That's in people of European ancestry. And 2 to 3% based on the available GWAS of the variance in hippocampal volume is accounted for in non-European ancestry individuals. Obviously, the push for diversity in the ADSP and across NIA in general will improve these markers. But it's very hard to dismiss that common genetic variation is influencing hippocampal volume when this degree of explanatory power is available. Nick Martin cautioned us that doing genome screening of brain images is not going to be any more efficient than doing case control genome-wide screening. And he was kind of correct. And so these are graphs of the power On the x-axis is the number of people in the GWAS, where 5 is 10,000, 6, it's on a log scale, 6 is 100,000. As more and more people take part in the GWAS, a larger fraction of the SNP heritability of each of these traits is explained, with cholesterol and other blood markers and analytes being very, very simple in their genetic architecture and easier to discover the variants that affect them. Alzheimer's disease being somewhere nearer the most efficient neurological disorders to study. And then as you go into psychiatry and then more complex psychiatric traits and conditions, very, very large samples are needed. Now, as you travel around the cerebral cortex, the common genetic variants that, again, affect each region of the cortex, here it's the thickness of the precentral and postcentral gyrus, is partially overlapping and partially distinct. You can imagine there's a sort of genetic modular architecture in which the common variants that we discover exert an influence in a somewhat restricted way. And in this paper in Science last year, many of you, thank you very much, contributed to this. A mosaic or roadmap was created with many, many loci in the genome that affect the morphometry of different regions. Are there any patterns? It's difficult to discern patterns. This is an ideogram of the genome and the loci that affect markers of cortical thickness and surface area. There are some hotspots. There are areas that tend to have variants that affect a very large number of cortical regions and traits. And we're just beginning to understand the association between these and the patterns that we're used to recognizing in the cortex. And if you plot the Manhattan plots for each brain structure, you can begin to see some modules and features that we didn't anticipate. There are genetic correlations. So the common genetic architecture that affect different brain regions can be clustered. And there are essentially sets of brain measures that tend to have genetic correlation and have similar genetic influences. And also the loci affecting brain morphometry overlap with those that are associated with Parkinson's disease and AD, mainly through ApoE and MAPT, because we know that they affect both AD risk and PD risk. And as it happens, they affect the risk, or I'm not going to say the risk, they affect the volume or morphometry of the brain. These GWAS are queryable in ENIGMA 3's browser, which you can look up online. I don't want to forget rare variants. So rare variants in the genome, they can be copy number variants, they can be deletions and duplications. Huntington's disease is a very prominent example. These tend to be so rare that they're difficult to study on our own unless we're a clinic that assesses people with genetic deletion syndromes. But overall, the cohorts that I've discussed, they were able to call their CNVs, very large scale projects to do this, and find that very, very large effects come from rare variants in the genome, up to 1 or 2 standard deviation shifts relative to healthy controls. And many of the rare variants in the genome that confer increased risk for developmental or psychiatric conditions, they're associated with a latent pattern of brain differences that is very, very similar to the idiopathic diseases that they cause risk for. And so this is, again, ongoing work. Ida Sonderby, who leads ENIGMA CNV, is making a dictionary or a catalog of the markers, the rare variants in the genome that affect the brain. Moving to function, you can do the same with EEG or MEG, and ENIGMA is certainly doing this. If you have a harmonized analytic protocol for EEG, you can begin to comb the genome for markers that affect spectral power, many other functional characteristics, brain synchrony, and so forth. And the genome-wide screens of EEG have actually found multiple markers that link to the frequency components of oscillatory brain activity. Some of them in the GABA neuroinhibitory pathway. Common variants affect both beta power in the brain, as observed with EEG, and also affect risk for alcoholism and epilepsy. The GABA pathway has a number of receptor subunit variants that affect not only risk for substance use and for seizure disorders, but also seem to affect this very clear and precise biomarker on EEG. Moving to epigenetics, there are many studies, this one led by Sylvain de Rivière at King's College London, that have searched in this case, the methylation profile of the genome at these CPG islands to understand if there's anywhere where the degree of methylation is linked, in this case with hippocampal volume, and there is. So this is more difficult to do than genome-wide screening because fewer sites have it, but this meta-analysis of 11 cohorts and a little over 3,000 people has found 2 loci where methylation of the genome is associated with hippocampal volume. Now you could say, well, the structure of the brain is interesting, but it doesn't really tell me about any ongoing processes. It's just a snapshot. So ENIGMA Plasticity collected all of the MRI data from people who had been scanned at least twice. And so in ADNI and CHS, many other cohorts that your ADRCs are developing, you follow up your patients many, many times. And using standardized protocols, Rachel Brower and Hilke Holzhoff-Paul, who led this study, computed the annual rate of volume change for hippocampus, for lateral ventricles, for many of the structures we care about. Now, these plots are not volume, they're the rate of change of volume. So on the x-axis, there's the person's age. And so as you see here, it's almost like the derivative or the time derivative of the measures we looked at before. And they found that in 10,000 people, again, aggregating data from over 20 cohorts, there were loci in the genome that were associated with a rate of tissue loss in hippocampus and other key parts of the brain. If you stratify this into aging and development, obviously there are very different markers that are relevant. And again, some of them are operating all the time, but others like ApoE tend to operate with an age interaction. So some of these markers in a straight GWAS will not necessarily come up because they may have a time of operation that is later in life, and putting the younger cohorts in there may obscure them. Now, this is all univariate, and there's a lot of interest in ENIGMA. ENIGMA is based on a code-breaking effort in the Second World War to see if we can do a better job of modeling genetic variation and its influence on the brain. So there's a great deal of interest in AI, a program, the ADSP machine learning program that's been championed by Marion Miller at the NIA, is looking at methods, both classical statistics and deep learning, that find the best linear or nonlinear combination of genetic markers that explain the brain biomarkers that we're collecting. And this is work that is also guided by bioinformatics work, AMP-AD, and many of you contributing to the functional genomics efforts, Alison Yao and others at NIA. Those data are being used to guide the selection of genetic biomarkers, because obviously it would be more beneficial if genetic markers were picked that were either druggable or lie in known pathways where the downstream characteristics on gene expression are understood. Another extension is to do brain-wide, genome-wide association. So you could certainly, in your PET data and in your MRI data, ask the question about voxel-wise associations across the genome. And now with the benefit of Arthur Toga and the Alzheimer's Association's GAIN project and all of the ADRC data sets contributing to NACC and other efforts, there's certainly enough statistical power to overcome the multiple testing correction that's associated with screening the whole image. And just to show you that this works, I mean, those top SNPs from the GWAS that we've been talking about, if you look in ADNI 1 and ADNI 2, and then Hunt and then the Queensland Twin imaging study, each cohort on its own has rather speckly maps of association, but when they are meta-analyzed, in other words, voxel-wise combination is used to, in this case, just pull data from 4 cohorts, you get absolutely lucid associations. I mean, this is the putamen. And so we wouldn't have known from the ADNI data alone, although it looks like a glimmer of evidence, that this genetic marker is just so extremely specific in its effect, which is, you know, one of the remarkable benefits of having a network like the ADRC network to club together to do this. Just changing topic to different disorders, we talked about genetics of brain. All of these disorders as well are having their brain imaging data meta-analyzed by ENIGMA. The idea is you compute brain measures again from the scans, but this time in step 2, you test associations between brain measures and clinical severity or risk factors or possible modulators such as treatment. And again, the evidence is analyzed across cohorts. And just to show you some of the disorders, these are studies in many cases of 3,000-4,000 people, in some cases 10,000 people. The cortical volume reduction that you see in each of these disorders obviously depends on disease severity and a number of other comorbid conditions. But at least to first order, you'll see that many of these conditions, neurodevelopmental and psychiatric, that have really led to a great deal of controversy as to what their neuroimaging correlates might be. You can begin to assemble, in a sense, a roadmap that, at least a first approximation, shows you the morphometric differences in the conditions. So, I mean, just some obvious comments to make. In major depression, the cingulate and limbic deficits tend to dominate. In bipolar disorder, a very large ENIGMA study of nearly 6,000 people, the frontal lobe deficits tend to dominate, which is consistent with our understanding of bipolar as having a sort of inhibitory dysfunction as well as a reward pathway dysfunction. So you can, for every disorder here, schizophrenia, bipolar, major depression, make a catalog of the brain biomarkers that are most differentiating of the disorder versus controls, and then for that matter versus the other disorders. And you can see, I mean, there's a very nice study by the Kukull Consortium in Japan. You can see if these hold true in other people of other ethnic backgrounds. So they at Kokoro have been running the ENIGMA pipeline on psychiatric conditions and finding that the rank order of brain correlates, in this case of schizophrenia, is almost exactly the same from Japanese cohorts to European ancestry cohorts. And as you know, NIA and NIH in general is doing a great job of diversifying the cohorts that are assessed in these types of studies. And just a encouraging note, the biomarker data coming from Japan, at least for the psychiatric studies, is extremely consistent with the data coming from the European ancestry groups, which isn't to say that they won't have differences at some point, but to first order, it's similar. Just to show how this is done, I mean, this is a study of schizophrenia. This is where the data are. The algorithms go to the data. So the data, because of data transfer complexities in different countries, the data is not typically transferred, but it's then analyzed and a forest plot is generated where every cohort says, how much difference is this study of schizophrenia in this brain measure between patients and controls. And you'll all remember the time when, 20 years ago in schizophrenia research, there was a great deal of disagreement as to whether there were any robust brain correlates of schizophrenia that could be found. But now these are very, very robust. And again, you can see from the forest plot, not every cohort has the power to find this effect on its own. And so there will still be some inconsistency in the literature about whether these things are always discovered. This can be done voxel-based. So as you add cohorts, this is a study of OCD, obsessive compulsive disorder. Rather than this notion that the effects are very limited in the brain, when you begin to aggregate data from here 31 cohorts, pretty much all of the brain is altered to some degree, rather subtly. There are ongoing studies of Parkinson's disease. ENIGMA Parkinson's was just funded by NINDS. And one of the things that we shouldn't overlook is obviously in disorders that are progressive, such as AD and Parkinson's. I mean, this notion of aggregating these maps doesn't necessarily make sense. It should be stratified by disease stage and for that matter by subtype. And so I think the next layer of studies going on in ENIGMA would not be looking just at case control effects, but also at stages and subtyping. So in the last 2 minutes, I'll tell you about an extension to Alzheimer's disease. So there is not an ENIGMA group that focuses on Alzheimer's disease. And that's partly because NIA has many, many initiatives, NACC and all of your ADRC centers are contributing to these, to do multi-cohort assessments of Alzheimer's related biomarker and imaging data. This new project, it's a UO1 as part of the ADSP machine learning initiative spearheaded by Marilyn Miller, is looking at whether or not you can go beyond meta-analysis. So rather than just meta-analyze a certain biomarker, it's much more complex than that. All of the genetic and omics, and for that matter, real-time data from patients would influence your predictions about diagnosis, and for that matter, prognosis and treatment response. So there's a lot of work, not just on classical statistics, but neural networks, deep networks in particular. Many of you are pioneers in this area. I see many of you on the call who have developed methods for this, to try and subtype the data, not just the imaging, but also the clinical omics data, according to the ATNV framework, according to likely to response to treatment. And many efforts in this regard have been very successful. There's disease classification methods using deep learning that have been applied to over 50,000 scans with very, very high accuracy for Alzheimer's classification. Work by Andy Sakin and Taiho Zhou using deep learning to discover unexpected features in tau PET that then can be targets for genomic screening. So you can imagine a sort of two-stage approach where you use deep learning on imaging to pull out highly associated patterns, and then you go on to use those patterns as a more precise target for genetic screening. And also through ROSMAP and other cohorts that have neuropath, there's a lot of interest here, work by Dugu Tosun at UCSF, where she's learning from in vivo imaging what the predictors are of particular neuropath subtypes, which of course may be overlapping, of course, for drug trial stratification and post-hoc analysis. So this again is a very, very big team effort. All of the ADRCs, as you know, have contributed and pioneered the collection of whole genome sequencing. And as you'll see from the genome-wide screens, it's now reasonable to search for rare variants and for that matter, whole genome-wide variants associated with the major MRI and PET biomarkers. We're not suffering from their statistical power, even though, as we said at the beginning, Nick Martin had a cautionary note about the power not necessarily being better for endophenotypes versus others. There's some exceptions, as you all know. There are some biomarkers that are more efficient to discover genetic variants. So just a big thank you, all of the people worldwide in 45 countries that have contributed imaging data to ENIGMA, including many of you who are on the call at the moment. If you want to join in, I mean, you can join ENIGMA at the website, at the Stevens Institute, at the INI at USC. You can go and have a look at that. And a big thank you to NIA, which I have to say kept ENIGMA going after the big data to knowledge funding elapsed. NIA is now funding ENIGMA and AA for AD. The Alzheimer's Association is funding ENIGMA's APOE2 project to look at the correlates of E2. And if you're interested at all in contributing, I mean, do reach out to us. And again, a big thanks to Marilyn Miller, John Shao, Brad Wise, Alison Yao, Jenny Larkin, who've really helped us champion these efforts. USC's ADRC, led by Helena Chu. And also, I don't want to say this is all ENIGMA. I mean, we're in doing all of these studies, we're benefiting from work that's been led also by Charge, Sudha Seshadri, Charles DeCarli, Claudia Sotisabel, and Oscar Lopez, and many of you that I haven't had time to mention who've made these studies possible. So again, thanks, very honored to have the chance to speak with you today, and thanks to Dr. Oscar Lopez for chairing this session. Appreciate it very much.
Thank you, Paul. This was very impressive. And we have some questions from Richard Caselli. It would seem that he says that amazing work, but with large enough samples, it would seem MRI differences can be found with almost any common disorder. So how do you choose your controls? Are the controls completely normal, or do they simply not have the specific disease of interest?
Yeah, thanks so much, Richard. I mean, as you all know, I mean, you have to be very cautious. So using, particularly in the psychiatric studies, people with no comorbidities whatsoever will give you one answer, is not necessarily going to give an answer that's specific to the condition that you're studying. So, as you all know, we would include in ENIGMA and beyond cohorts with comorbid vascular disease. We might include them or not. In our GWAS, we have healthy only. And also two other cohorts, one called neurohealthy. In other words, they may have other conditions that don't necessarily affect the brain. And then neuro comorbid, where they might have the condition of interest or not. And as you all know, I mean, we have to not be misled, particularly in these very highly powered studies, that the differences that we find are attributable only to the covariate that we care about. And I think, all of you have contributed to modeling these other concurrent disease processes with very, very high power. And I think it's a very good cautionary note to make sure our controls are stratified in multiple ways so that we better understand the specific differences for the disorder.
Okay, I think we have one last question from Scott Small. Fantastic talk, Paul. In the changes observed over the lifespan, are you trying to exclude common age-related disorders like Alzheimer's disease to try and isolate aging per se from age-related disorders?
Yeah, a great question, Scott. And you, as you know, are doing great work in AI in this area. I think, again, the answer is to split the data multiple ways. So obviously, as you see in these lifespan curves, if you only restrict your attention to healthy aging populations, you get this anomalous increase where the super healthy people are the only people remaining in the plot, or for that matter, they're differentially represented late in life. So I think, and you know the answer to this, using multiple references that don't exclude people with age-related conditions as well, will give us a better picture, not just of the genetic variants, but also the modulatory factors that are relevant. So great question.
Thank you, Paul. Excellent presentation. We're going to move to the next presenter, Andy Singleton from the NIA. He's going to talk about the card.
Great, thank you so much. Okay, that was a tough act to follow. Every time I see Paul talk, it's pretty mind-blowing, but that was really incredible. And I will say, just seeing what's been done in the ADRCs has been just incredibly educational for me. It really is an amazing achievement, and it's great to see how closely the centers work together. So the first thing I'd like to say is thanks so much for giving me the opportunity to talk about CARD, the Center for Alzheimer's and Related Dementias. This is something I'm incredibly dear to my heart, of course, as the director and as someone that's been involved in the inception of CARD really from 2018, so almost 4 years ago. This is a joint NIA and NINDS initiative that sits within the intramural research program of the NIH. So we know this, of course, the US government has really provided an incredible amount of support and an incredible opportunity for research into AD and related disorders. And without question, the engine for discovery is going to be this group and other extramural investigators, right? The large proportion of funding is going to the extramural world. That is the engine for discovery. But as we thought about this opportunity within the IRP, we started to think about how could the IRP contribute, right? We could, of course, just create a series of labs and contribute in an additive way to the field. And I think that would have been fine. We thought that there was an opportunity to really start to take advantage of some of the things that are different about the IRP compared to the extramural world. And this really led to us thinking about this structural opportunity. The IRP is certainly different from the extramural world in several ways. And I think that we need to really take advantage of those differences as we start to think about executing science in general and executing science in the space of AD and ADRDs. So of course, those are obviously that the IRP has stable and generous resources. We have secure employment. Importantly, our review is not entirely prospective. So we have the opportunity to execute science very quickly and then present it at a later date. And there is, of course, the potential for us to define our own metrics of success. So we don't necessarily have to aim towards the same endpoint that everybody else has to. So in thinking of these differences of the IRP compared to the extramural world, we started to think about, in creation of CARD, what kind of program could we devise that would take advantage of these differences, where we could perhaps contribute significantly in a slightly different way to the ADRD field than the engine that is the rest of the extramural world. So this is what we came up with in terms of a programmatic strategy for CARD, which at this point, as we started to come up with this strategy, was really still just a concept. So we thought that it would be incredibly important to focus on addressing critical barriers to progress that are not easily addressed using existing mechanisms. So what does that mean exactly? What are those barriers? So here I'm thinking about things that are rapidly emerging opportunities. So of course, an issue that you have, or a challenge that you have in the extramural world, is that as opportunities emerge, you have to go through grant cycles to propose experiments and to move forward. And we can, of course, move quite quickly in that space. So I think taking on challenges as they emerge and those that could benefit from really quick, nimble investigation. The converse is true as well, in a way, in that because we have very secure resources, we can also take on projects that require an incredibly long-term commitment without having to worry about continually having to compete for additional funding. A space that I think is particularly dear to my heart, because this is a space that I've worked in for a number of years, is working in a space of science that's centered on addressing practical barriers. So those that don't necessarily do particularly well in grant competition, things that are centered on platform production, building infrastructure, capacity and capability building in different populations and different fields. Of course, areas that are incredibly collaborative and cooperative, particularly when you're working across domains or working across types of research, be it from industry, academia, stakeholder organizations. When we started to think about execution of these types of projects, it became really, really important to us to really focus on a program that was one centered on an extremely mission-focused and program-focused approach with very clear go, no-go points, milestones, deliverables. So a very kind of tranche-based research model rather than the one that is in a typical kind of academic research model. Key to all of this, and why I'm so pleased to have been invited to talk today, is engaging and understanding the needs of the research community. I think that for us, everything we do in CARD, we're aiming to complement ongoing research activities. Again, the work that's being done in the ADRCs, the work that's being done in the extramural world is the engine for discovery. And our hope is that we can just help a little bit in making that engine as efficient as it possibly can be. So in the context of CARD, as we moved through deciding how we wanted to execute science, where we thought there was an opportunity for us, the next part was deciding what kind of science we wanted to focus on, at least in the first instance. So we have some clear scientific themes in the center. The first is molecular pathogenesis anchored in genetics. Perhaps no surprise, I'm a geneticist, so that's probably not a surprise to the rest of you. Focusing on disease subtyping, prediction of risk, and progression and prognosis of disease. Filling in this gap of de-risking therapeutic targets, so taking them forward from the stage that we usually leave them, where it's a target and you just hope that a drug company picks it up, to working carefully with industry to try and figure out what are the things that you need to start to move these targets forward. What can we do to reduce the risk and make these targets more attractive, not than other neurodegenerative disease targets, but than other targets across all diseases. And then lastly, I think NIH has had a storied history in trials, certainly in genetic trials, a very early history in genetic trials. And I think I would like to see us continue to work in that space, working in the space of development and application of precision therapeutics, likely in the monogenic space early on, and then moving into complex disease. As important to us, or to me, as those scientific themes are these structural priorities. So we have a really large focus in absolutely everything we do on diversity in research and researchers. So really leveling the playing field for researchers from around the world. Part of that is democratization of data. Part of it is also transparency and reproducibility. As I mentioned earlier, I think collaboration and cooperation will be key for us. And above all, as we start to think about the products that we produce, they have to be actionable. So they have to be useful to the broad research community. And I'll talk about a couple of those later on. So how do we actually do this? How do we affect this? So we came up with this strategy, which initially starts essentially with an idea factory, where we're bringing in ideas from investigators within the intramural program, extramural scientists, investigators from the extramural world, academia, stakeholder organizations, to help us identify ideas and barriers to progress in the ADRD space. Again, fitting within this a rubric that I described earlier, the key kind of approaches that we've prioritized. And we go through this process of due diligence, essentially, where we take the problem, take the idea that's been put forward, and understand where does the field sit currently? What's the problem scope? What's the knowledge state? Does this fit within our mission? And as importantly, does it fit within the program of CARD? Go through this circular process to start to develop ideas and come up with a project or program or research to move forward. This is then followed by this, again, kind of tranche-based research approach, where we don't really give money to an investigator within CARD and say, okay, come back in a couple of years, let's see what you've come up with. But we're really focused on a very milestone and deliverable focused approach, where we source solutions, and those solutions can be internal work within CARD, can be collaborations with CROs or with academic labs or with industry, and really follow the project through. All of our projects are managed by professional project managers so that we're held to our milestones, held to our progress, and have clear go, no-go decisions as we move forward. So as you think about a center like this, right, it's not a typical academic center, and it would be very difficult to have people that are there on an academic track staying within the center forever, because of course, you can't really have the same academic freedom in a very mission and program focused center such as this. So we have a kind of permanent operational structure, which is the overall leadership for CARD, leadership for the basic and translational and clinical domains, and program or project management staff. We have some permanent expert groups. So these are groups that we're building now that have expertise in various scientific or clinical approaches that we think are particularly important for CARD's program. Then we have a series of term-limited appointments. So the ones that we've used at the moment are mostly internal. So existing NIH investigators who are whose permanent position is in another lab come and spend time at CARD using CARD resources to perform and execute a term-limited project. We hope to float the same for extramural investigators, either as collaborations or as visiting appointments within CARD, the same for industry scientists. We'll have a very small number of tenure track investigators who work on an extremely CARD-focused program of work for the duration of their tenure track, and then continue in the AD space, likely in other labs within the NIH. And then a new program which we just launched, which is the Independent Research Scholar Program, which I'll talk about in a minute, which is aimed at creating a new generation of young scientists. So the Independent Research Scholar Program is one that is being launched in the next 2 or 3 weeks, called the ARDIS program, Alzheimer's and Related Dementias Independent Scholar Program. And here the idea is to provide independent resources for early investigators. So scientists who are only 3 or 4 years, less than 4 years out of their graduate work. They come to a CARD and are given independent resources for 3 to 5 years, pretty generous resources and access to everything that's going on within CARD. These positions in particular are reviewed not only on scientific excellence, but on a commitment to diversity. So a commitment to diversity in the research that the investigator is performing, but also a general commitment to diversity. So I think this will be a really interesting program as it launches in the next couple of weeks. In terms of getting, I'd shown this due diligence space earlier, where we're challenging people to come up with problems or ideas in the AD space. And this has just been launched within NIA and NINDS, and will be launched out to the more broad NIH community, to all intramural scientists and extramural staff. And essentially, this process is one where we take a very broad concept in the early submission stage and work with investigators to really develop this concept and come up with a program of research, assuming that it fits within our program and our mission, that can then be executed within CARD. So this is the formal program that again was launched a week or 2 ago. Informally, we've been doing this within NIH, and this has led to a whole series of initial work packages. I'll talk about a couple of these. Again, these have involved internal scientists, but also collaborations with folks outside of CARD, many of whom are likely here today. So the India initiative is one that touches, I think, really in some way all 4 of our scientific priorities, and certainly many of our structural imperatives. So this is the iPSC Neurodegenerative Disease Initiative that's being run by Mike Ward and Mark Cookson. And the aim here is to create a resource for the research community to in the space of IPS lines. So I think one of the challenges that we saw early on, certainly in the disease space is that we see a huge uptake in iPSC work, but also a really varying degree of standards and approaches in iPSC work, and lots of N of 2, N of 3 papers being published, different lines, different backgrounds, making it extremely hard to actually compare data. So our first step in trying to tackle this problem was to create a uniform set of cells for the research community. So here, focusing on ADRD in the first instance, taking 73 genes, 134 different variants, and creating on the same background a single nucleotide variant knock-ins, revertant lines, homozygous knockouts, tagged, halo tagged lines, all immediately available to the general research community. So this is phase 1 of the project. The second phase is then molecular characterization of these lines, all of the things that one might expect, omics, imaging, and data release. So as I mentioned, we're still in phase 1, generation of the isogenic lines. This has been done in collaboration with Jack's Labs, Synthgo, and Thermo Fisher. Again, coming back to this operational structure that I talked about earlier, where we outsource things to CROs where necessary. So this program is only a little over a year old, but I think it's been incredibly successful. It's not even at the release state. It's not really been advertised, but we've already distributed 143 indie lines, shipped to 56 different investigators in 10 countries. I think it's, given that we've not even advertised this yet or made the public broadly aware of it, I think it's been incredible. All of the indie plasmids are on Adgene. The trio sets will start to be distributed fairly soon. So I think this has been a great success. The other part of it that's been a success actually is that we are, of course, focused on ADRD, but other disease groups are contacting the Indie Initiative and saying, we would like to mirror what you've done so that we can then compare across diseases. So additional lines have been created by support from ASAP and CZI. So you can see we've gone from 150 different variants to 189 variants now. I think we have over 800 daughter lines being produced now. So it really is a pretty major initiative. Another major initiative within CARD is around data science. Again, this touches every aspect of what we're doing, and I think all of our strategic priorities. We're really producing here in CARD a product set that is aimed at addressing a few things, really. I think it's aimed at reducing the activation energy to access data and making data more broadly available, and also encouraging collaboration across labs. So we have this series of products that are being produced by the data science group that range from a data lake, so a space in which all data is brought together, harmonized, and is analyzable, all the way through to smaller tools like ClusterBuster. The earliest of this actually is kind of bouncing on from the last talk, from Paul's talk, is an automated machine learning package. So this is essentially a machine learning package that tests and picks models for you based on the best performing model, particularly focused for genomics data. This now has 16,000 users. We released this about a year or so ago. So it has 16,000 unique users currently. So I think this has been a real success. And as these other products start to roll out, I hope that they will also be as successful. So how about the next stage, right? We are, of course, developing. CARD is pretty young, pretty new. The next stage is starting to engage with academia, industry, and other stakeholder organizations to get an idea from you all. What are the things that you would like to see done that aren't being done? And is there a space for us to help? Of course, this is why coming to talk today is great. We've already reached out to a lot of labs in a fairly ad hoc manner, based around research priorities that we started to scope out. But I would really love to have many more conversations, particularly with the ADRCs, to really get an idea about, is there a space for us to help? And I think there will be. particularly when you think about the types of projects that we're game to take on. So is there space to help in the execution of projects, or are there resources that you would like to see produced for the general field? And we'd love to have those conversations. So as I mentioned, the original concept for CARD was almost 4 years ago. We are building a building on the NIH campus, which will be finished in February. So from concept to building, we'll be moving in February. It will be exactly 4 years. So I'm really excited. Once we're in, we'll actually be able to really start rolling. Everything we've been doing so far has been without permanent space. I'd also like to highlight we have a few searches ongoing, Director of Clinical Translation, a tenure track position, the IRS scholars that I mentioned earlier, and some staff scientist positions. So with that, I will stop. And again, thank you so much for making the time to listen to me today.
Thank you, Andy. Very interesting and innovative project. It has a huge potential to potentiate synergism with the centers. And I think that's is going to happen. We have one question for you. Is how does one find out about resources that can be obtained from INDI? Is there a web link? Is there an automated request form?
Yeah, there will be. We are. So the website is being built at the moment. There's a kind of there's an early stage website that's available. But the best thing to do is to reach out to me or Mark Cookson or Michael Ward, and we can put you in, help you access those lines. Our advertising and outreach campaign will start in the next 6 months or so. Again, we're still kind of in the early stages.
Okay, well, thank you very much.
Yeah, pleasure.
I think that we can move to the next speaker, Dr. Thomas Karikari. He is in Sweden and is in his way to Pittsburgh. Thomas.
Oscar. Thank you very much. I'm going to share my screen now. Thank you. So
we don't see the screen now, so you can upload.
Let me try again.
Oh, okay. Yeah.
Thank you very much. So I'll be talking today about my synthesis on the future of blood biomarkers. As Oscar said, I'm on my way to Pittsburgh. I've been training here in Sweden with Drs. Kai Bleno and Henrik Zetterberg. And I'll be starting a new life in Pittsburgh, and I'll be joining the ADRC there. So it's really exciting to be initiated into the AD community in the US. So what I've been trying to talk about today is that we know about all the recent big buzz about biomarkers and blood, and how exciting it is that we could transition from CSF and PET into these new markers. But then there are some fundamental questions that we need to ask, and these have to be addressed. And I feel that one of the best ways to look at these is the power of the ADRCs and the communities that you do have here in the US can help to address. So it's more about raising some unanswered questions, some open questions, and we can all try to think about those in different ways. So I'm not going to bore you. AD, as we know from a pathophysiological perspective, is got amyloid, tau, and neurodegeneration, and that we got markers from imaging perspective and CSF perspective also. And more recently, we seem to have the full spectrum of the ATN markers in blood as well. But then the question here is, how do we get to use these markers in the best way, and how could we transition them into the clinic, and would they be able to replace or support what we do have in the clinic currently? And when I talk about blood biomarkers, I tend to always come to this figure because it's one of the, I think, one of the exciting ones. For example, here, showing that blood biomarkers provide AD neuropathological evidence. That, for example, this is a cohort from London that had provided quite unique because there were neuropathological evidence at the end of life, but then they provided blood samples around 8, 4, and 2 years before death. But then when you measure plasma phosphatase across those time points, and then you try to integrate with our clinical diagnosis, it didn't perform well. So the diagnosis data that we do have here is the AD in red, non-AD in green, and control in blue. But then when you split that to the form of, if you do it according to neuropathological diagnosis at the end of life, then it becomes to make sense from a biological perspective. So here, starting to show that AD has biomarkers, especially here, because at this point it was the only one we had was phosphatide 1A1, provides evidence from a biological perspective. But then it doesn't seem to integrate too well with clinical diagnosis. So this is both good and bad, because from a pathological perspective, we could say that what we're measuring in blood provides information that relates to the biology of the disease. But then in real sense, we want to use these biomarkers in the clinic. And then if they are not integrating so well, then there's a lot that needs to be fixed. So yes, just trying to focus the talk today quite more on phosphatide because it's one of the newest. But then just saying like there's a lot more biomarkers of phosphatide that continue to become available. And what are the benefits that we could have from blood tests? We've talked about these over and over, but just to recap, we could then be able to confirm diagnosis of AD, assuming that it's always clear-cut, that if someone's got AD, dementia, they don't have the massive increases, and those normal controls, we have nothing. But in a clinical sense, I guess we're going to have some in-between that will make the situation a bit murky. And then differential diagnosis will also be very, very important here between AD and some other dementias. Again, here you could have some with co-pathologies that you would have to deal with as well. So it makes it less straightforward. And for screening purposes, we have these classical Jack curves, and they're really beautiful. But I keep saying that these curves have been prepared from very well-defined cohorts that we have studied. They are cohorts or they are populations that we do not know anything about. So the question is, do these curves and these trajectories, do they hold universal, or there are some caveats that we have to be aware of? And that there's been a lot of talk recently about clinical trials and enlarging and making it more diverse. And this is one of the beauties that I think blood could provide, among several other things. So how could we use it in the clinic? These are the questions that I think everyone is trying to answer now. And these are our synthesis, what we have talked about here. What we try to suggest is that blood, PETA, for example, or the other blood biomarkers would not be used or should not be used as an isolation, because with all the insightful data being shown, it seems like once you measure PETA, boom, you're going to see everything. But no, it should be integrated with the regular clinical algorithms, the clinical procedures. So for example, if someone comes with a clinical complaint, then the normal clinical interview, everything that gets to be done should be done. And that if there's a suspicion of AD, then you could run, for example, PETA as a first pass, and then you would do the normal neurological exam, and then the cognitive screening all gets to be done as usual. But then we propose that if PETA levels do vary, for example, then if we get to have low, medium, high, more like a traffic light approach, and if you integrate that with the neuropsychological results, then you could begin to identify who to refer to a specialist care. It is still not as clear-cut as is being proposed. For example, how do you show that someone is got low, someone's got medium, someone's got high, or someone's got abnormal PETA? Because what is abnormal? And these are things that I think we need to discuss. So start with, what does increased PETA even mean biologically? And then we could link it clinically. I mean, there's been several results and several really exciting papers, but I just chose this to show an example. For example, in a recent paper that we showed in ACTA, that when you take A beta pair and then you split it according to quartiles, that phosphatide 231, for example, there was non-overlapping 93% confidence interval across the different levels. So then, but then when you compare, and it appeared to perform better than the others, but then when you compare with, let's say, tau PET, there was a lot more overlap. So could you then begin to argue that phosphatide, it's more of a measure of amyloidosis instead of tau comparatively, because amyloid often comes in before tau? And this figure from Nicholas Matson comes quite nicely, where they suggest, using some really nice data set analysis, suggesting that A-beta deposition leads to phosphatile release, both soluble in CSF and in plasma. So if this is the case, then clinically, should we then argue that if you find someone with an increased level of phosphatile, maybe we shouldn't say AD, because if it doesn't agree with the clinical diagnosis, then that is a problem. Could we then begin to argue that maybe that is amyloidosis?
Ira, it's Angela Taylor getting back to you.
According to the ATN system, it is AD pathological change. And then that could, for example, suggest preclinical disease, and then we could take it from there. And we don't know how Peter is going to perform clinically. I know of several cohorts and several studies that have been done in a real-world setting, but these are the few that were published recently that from our groups, and just seeing that maybe as a snapshot, how would this look like? And we shared for 101 and 231 in separate papers. But then if you take as a primary care cohort, for example, and these are individuals who have received, preliminary diagnosis in the primary care cohort, they haven't been confirmation in specialized settings. And if this is the profile you see, luckily in young individuals, the levels tend to be lower. It's a bit more convincing here. But then if you did not have these classifications, because these are even temporary, and this is what you see, to me, everything looks the same, right? So if this was the first pass, the first thing you use was phosphatile, and this is what you see, what does it suggest? How, as a clinician, what would you say? Well, potentially, and this is my suggestion, you could, assuming that we had a very well characterized cutoff, let's say that if that was a cutoff, then you could say other people that are above that cutoff then have to go on to receive specialized attention. And question is, how do you establish that cutoff, and how does that work? How would that work across different populations? And as I said initially, what would you call those? Would it say AD or amyloid changes or amylopathological change? And how would that integrate with clinical work? So here's my proposal. What I think is that probably if we were to look at increased phosphatile as a first, and this is a simplified way compared to the initial one, because I want to take it much easier, but this would be integrated with everything, so not in isolation. But if we're focusing more on the blood phosphatile, and if we were to find an increased level, then you could suggest that this is changes from an amyloid perspective. And we look at how could that really sync with the clinical diagnosis. And then from a biological perspective, how would that also work if we were to confirm with CSF or PET or neuropath, which happens much later. So I'll try and take these in turns. First thing, what makes blood phosphatile abnormal? If we want to say an increase or a change, abnormal changes, how do we define it? At present, we've shown those in cohorts, much different cohorts, and these cohorts are quite heterogeneous because the cutoff from one cohort does not necessarily apply to the other one. So how do we look at it? So to identify what abnormal phosphatile should look like, I'm trying to look at things from like a clinical and then an analytical perspective. For example, how does it change? How does a biomarker change across the life course? We don't know. We know that we've been able to study age in cohorts much well, but then if we were to understand a lot more what happens earlier, then we could tie together the sequence of what changes, and that could let us know if age adjustments may be needed, or if what implications will be for those with, for example, early onset AD. And just to say that, which is one thing we tend to forget, but phosphatile, or even tau in general, has other functions apart from AD, and one of those is developmental functions. And that's one of the data that we haven't published, but we find quite intriguing. This was total tau, but we've seen quite similar with phosphatile, that in umbilical cord, so those in babies, who have not been born, you get high levels of tau. It really drops in teenage, and then it goes down even further later. That the argument is that it would come up a little more when someone is diseased. So if we have quite a well-characterized understanding of what happens with these biomarkers during normal aging, then that would help us to understand what happens during disease. And that what factors would affect these measurements. Would it be factors such as sex, genetics, in terms of genetics, There is, in some cohorts, you see ApoE, E4 allele carriership has an effect, others you don't see. For example, this was a Down syndrome cohort that tend to see show an effect, but then just at a group level, but you see kind of a total overlap. And ethnicity, we do not know, and I'll talk about that just in a short while, and other comorbidities. Could these affect the levels? And neurological conditions, for example, we now know that, for example, if someone has a cardiac arrest, those with TBI, there are conditions that lead to acute changes in phosphatile levels, and these tend to clear quite quickly. So then we need to start thinking of all of these as a system, or as connected pathways that have to be. We can't take that once someone shows increased phosphatile, then voila, they should be having a disease process. And factors like cardiovascular, we do not know much about. And then lifestyle factors, medicine use could affect it, and any condition that affects the metabolism of tau from blood. Because the difficulty is that what we are measuring in blood is a combination of the production of tau into blood and the removal of tau from blood. We need to have a good balance between these two. Anything that tilts it in the other direction gets to affect the amount of what we measure and the trueness of what we measure. And some of these could be like liver function and kidney function. And Professor Michelle Mielke showed some of these data recently, suggesting that we need to be a lot more cautious in how we interpret these data. So does increased phosphatile for someone who comes to the hospital with concerns always mean AD? This is a recent data that we have in a cohort from Slovenia that we show that we looked at CSF, and interestingly, it appears that if you measure 231, 217, 101, you tend to see like in 231, CJD is not, this is control in blue, AD and CJD, and you get to have lower levels. But then in 217 more especially, we couldn't see a difference, we couldn't separate it. And that, interestingly, the CJD subcohort went on to have postmortem confirmation. And if you try to look at those who were diagnosed with CJD mixed with AD and those with CJD with no co-pathology, then it tends to be that 217 did not perform well. The question is, does this make 217 a bad biomarker? The answer is no, because this is something that is occurring independent of AD in a pathophysiological process that is not linked to AD, because what we saw was that 217 was the only one that correlated with total tau, for example, in the CJD subcohort. And one could argue that, well, CJD is a very rapidly progressive disease, so maybe that would not pose a much clinical concern. There's this recent paper where one of the mutations in the tau gene called the R406W, where it leads to a change, that it can also lead to increases in phosphatile 217 without a myelopathology. Again, just saying that we have to be more cautious and more integrative in the way we perceive phosphatile going to perform in the clinic. So let's come back. One of the things that could affect phosphatile performance is ethnicity, but we do not know much about. And this is the first data, and I was really excited to work with Susan on this. And so Susan did a very clever approach. We got some CSF data, but they do not, the individuals from both sides, so those, for example, with non-Hispanic whites and African Americans or other groups are not matched. So you could have differences in age, differences in sex, differences in cognition. So then it doesn't make it really like for like. So what she did was to use a machine learning approach and selected in the WashU cohorts that they have those who were paired for all of these. So those who were paired for sex, age, cognition, and ApoE genotype. So it means that it was like for like, the same individual matched with someone with these functions, which makes it bring it ahead of what we've seen before. So what we saw was that CSF A beta ratio, there was a difference, which validates what's been seen before, that there was a less likelihood of CSF A beta abnormality in African Americans in this cohort. We saw the same thing for the in plasma A beta, and in a myeloid PET also there was the same issues. So which tends to be quite confirmatory that could it then suggest that the abnormality of A beta does vary across populations, at least these two populations that were studied? If you look at this, the phosphors, CSF phosphatide 1A1 showed a difference. This was with the Lexis platform, but plasma 1A1 did not show a difference. And I'll come back to this quite soon. These were both 1A1 assays, phosphatide 1A1 assays, but they were not identical. And I'll come back and talk about that soon. And phosphatide 231 also the same, did not show a difference. However, in logistic regression models, what Susan found was that if we were to predict CSF A beta ratio abnormality using A beta 42/40 ratio, then you don't get to see a significant effect of race. But then for 1A1 and for 231, then you would have to see, you would see significant effects, which means that you might have to correct or adjust for race if you're using these biomarkers to predict for amyloidosis in populations, including African Americans, as they suggest. So there's a lot more that we need to think about. And the question is, what do you think about other populations that we've got no idea about? They're like, when I do a crude approach, if you base on Europe and America and maybe Australia in terms of population, maybe it's about 20%, and I would argue that about 4/5 of the global population do not have access. So maybe there's a lot more we have to do, and this does not only include those outside these continents, but even with these continents, we know that it is not everyone who has access to these established methods. So now I'll talk about the analytical aspect, what makes plasma phosphatide abnormal. How do we define abnormality from an analytical, the analytical factors that can affect it? So I've talked about non-clinical. For analytical ones, that could have some pre-analytical factors and variability. I won't bore you with these words. I'll just try and bring you home so that we can understand it. And the effect of these ones is how they affect the robustness, that how precise these measurements can be all the time. So for example, we did something where we plotted the density of these values for CSF phosphatide, CSF A beta ratio. Those were positive, those were negative. And then we did the same for 231, for example, phosphatide, and they perform really well. That, you know, we get to see if you have the line of abnormality using a cutoff, they perform pretty well. That quite well, good separation. And that when we looked at plasma, for example, the A beta ratio, the plasma A beta ratio did not seem to, so in terms of AUC perform quite well, but then it would suggest that if there's any tiny things that would affect the analytical performance, that would shift the bias in any way, could affect how robust the biomarker is across different, from day to day, for example. Because for a biomarker, you need to ensure that for a robust biomarker, the room of change or bias from day to day, from person to person, from instrument to instrument, should be quite small compared to the massive room of the biomarker abnormality or the biomarker performance range. And these are things that we also have to think about once we want to go to a clinic. And this is simulation analysis, sort of buttressing what I just talked about, that, for example, if you randomly vary the day-to-day variation of the biomarker, then if you look at this from these biomarkers, then there get to be massive differences. So for example, you could have some, like, you know, for GFAP, for phosphatide, they were a lot more robust, and there are others that were not that robust. So from pre-analytical conditions as well, what are the things we have to look at once we move to widespread use? How studies are designed, how samples are collected, what kind of tubes are used, and then how they are even processed. For example, are they hemolyzed? What tubes do we use, and are they operated on ice for how long? What's the time between collection and storage? And then how do they get to be moved to the analysis center? These are things that we have to think about. So there's just data beginning to emerge, but for pre-analytical conditions, this is one of the examples that we have. We looked at 4 different tube types recently: EDTA, plasma, serum, lithium, heparate, and citrate. Basically the tubes that you use to collect the blood, and then what sort of additives that you would have in there. And what we did find was that the absolute concentrations from these colors refer to the same individual. So if you collect at the same time from the same blood draw and you just drop the same volume into different tubes at the same time, what you get to see, for example, if you use the green, in EDTA plasma, the levels were quite high, but then it drops quite drastically in serum. It goes up in lithium heparin, and then in citrate it comes down back again. So it would be important which one that one uses and how that has to be used across. Freeze-thaw cycles also tend to have an effect over time, quite small changes, but that could be significant. And there are several things that we still do not know about. But if anything, the recent pandemic has shown us that we cannot always think about the conventional approaches of collecting blood and processing blood and keeping blood, right? And there are also then remote settings and where places where things will not be normal. So we have to think about different ways. And we try to experiment this lately, where we took these Noviplex cards, which are just a commercial card that blood gets to be separated whilst you spot it. You just take the blood and you pipette it onto that. The separation occurs in that. And then you could do a normal dry blood spot. You just take the blood and then you drop it on filter paper, let it dry, and later you put in buffer and then you dissolve it. Or if you were working just to mimic field work, if you delayed the centrifugation, so you just collected the blood, you stored it at room temperature for 2 days before you brought it to your clinic, and then you centrifuged it and processed it, what would happen? And for our first pass, we tried with NFL. And what showed was that you tend to have quite a pretty good correlation, like almost 1-to-1, with if you use the Noviplex card, if you use the dry blood spot, and also if you delay that. It may not work for all biomarkers, but at least this tend to make us start to think about different ways that we might have to start to imagine blood collection in different settings. So, amyloidosis. How do we define amyloidosis? Do we use CSF? Do we use plasma? Do we use PET? And how does that affect them? Does the choice affect the predictability of plasma PETA, for example, the ability of PETA to predict amyloidosis? And I think so this is a preclinical cohort, the alpha cohort in Barcelona, where we looked at recently, and it does suggest that for AUCs to predict amylopositivity from CSF and plasma phosphatiles, you tend to see, for the same biomarker, better performances when you use CSF, when you use A-beta PET compared to CSF, which would argue that it depends on which one you use, maybe especially the preclinical assays affects what you use. And how would this impact clinical decision? I think this is a big question, and I try to think about how we could solve this puzzle in a way. So, for example, if you measure in a simplistic way phosphatile and then a patient with cognitive changes, there are 4 different things that could happen, right? In a preclinical sense, you have someone with increased phosphatile, but cognitive sense they are fine, and that could be a discordance between phosphatile and cognition. In late dementia stage, also you could have cognition being quite poor, but then phosphatile starting to drop, because we tend to see in the few longitudinal studies we've studied that once you have extensive degeneration in late dementia, you start to see dropping levels of phosphatile. So how could that also work? Of course, in the regular sense, in a simplistic way, we will see very matching increases in phosphatile and dropping in cognition, and in normal aging, it would be even much easier to spot. But then, how would this work in a real world setting, and what would be some of the changes, the effects that we see, the problems, and how would we address them? So I'll finish that with confirmation with CSF and PET. So if you look at plasma phosphatile and CSF, how do they agree on an individual basis, not the group level? In this paper, in this data from Sebastian, this was not logged, so it's quite difficult to decipher, but then you get to see some reds, some pinks here, and then you also get to see some pinks here, arguing that you would see some individuals that do not agree from the phosphatile, CSF, and plasma, even for using the same biomarker. Right? And it's even worse when you use different biomarkers. So here's CSF phosphatile 181 measured with the Lewy platform, and this is the plasma phosphatile measured with the Seymour platform. And I'll come back and show why they do not agree too well. At in the group level, it's reasonable, but then at the individual level, you get to have these sort of 4 boxes, some belonging here, some belonging there nicely, but then there are others that do disagree. And because if you want to confirm, then this is important, because if you measure phosphatile and it's increased, and then you go to confirm with CSF and it shows something else, how do you explain that? And just another plot to argue that. But then someone might argue that, well, you guys have been showing recently that, for example, you don't even need CSF or PET, because phosphatile in plasma, especially 217, as I was showing in this paper, could do the job. It's performed just as well as CSF. Well, yes and no, because my argument would be that if you compare to CSF and plasma, you would see that increases in plasma phosphatile and then are the same for a similar trajectory for CSF. You see increases in AD, massive increases, and then very low levels in those without AD, even with other dementias, and then those with normal controls. But then when you get to see the MCI and those with different forms of cognitive complaints, then you get to have quite some difficulty. Those with A-beta positivity, even the gray zone, and those with heterogeneous groups, then that will be the place that we might have some problem integrating CSF and plasma phosphatile or exchanging them. Then I try to think about what affects interchangeability from the perspective of biochemical. So I try to plot all the biomarkers that we do have now from plasma phosphatile and also CSF phosphatile. And this is quite important because this is where the immunoassays do target. The classical CSF ones, they target these really short middle portions of the protein. Some go here, the others are much shorter. The blood ones go much longer, but they all go towards the end terminus. And from the beginning of the discovery of this, the phosphatile methods that we have, I was really excited when I did find out that it has to be the end terminus that you find CSF, you find phosphatile in blood. And by all these assays are targeting there. Now, it is important. What it means is that, for example, if you had let's say someone has a phosphatile, like a fragment here, but still phosphorylated there, this assay would not pick it. All the assays, if you stretched it, they wouldn't pick it. The CSF would pick, but this one would not pick. It's actually the same way. If you had those that stretched this way, the plasma would pick, but the CSF would not pick. So even though they are measuring the same phosphorylation epitope, they have some classical or some biochemical differences that we have to think about in the way that we explain the data. And why is this important? Is because tau in the brain, for example, there's a lot of truncation and different post-translational modifications, but you get to have the full complement. In CSF, from the data sets that we have, you tend to have some shedding of the C terminus and even more shedding in that. So it means that, for example, if you had a CSF assay that targeted this region, you may not have it working in blood. And that means that if you had a biomarker that was only a disease situation that was only showing this increase, then it might not work with blood assays. So what about phosphatile in blood and tau PET? Well, there's been several studies, and this seems to be quite more clear-cut, that you see phosphatile increases, but tend to increase before tau PET becomes abnormal. There are several studies arguing that. And I like this saying that is in plasma and in CSF we get to see what is actually happening in the brain, an indirect measure. But then with imaging, it's more about what has happened, because you have to have a build-up of aggregate over time before you can get to measure. And this can affect the interchangeability as well. And this is my hypothesis, that we have soluble p-tau in CSF and in blood, that we show that they are associated with pathology, the same way tau PET, for example, is associated with pathology. But then we normally see modest correlations, which argue that they're measuring the same thing but different things. And what I remember some of the classical old papers where they argue that when you have tangle formation, there's some phosphatile forms, when the pre-tangle stages, they are increased, right? The levels are increased, but then they drop once you go to the intracellular tangle stage. Others pick up at the intracellular tangle stage, but they just stay there. There are others that pick up when the tangles are formed intracellularly and also keep increasing when they go extracellular. So it means that if you're using biomarkers that are targeting these regions, these different phosphorylation sites, then they're going to perform differently when you correlate with aggregation. And I think this is what we might be finding. In terms of interchangeability of plasma biomarkers, p-tau biomarkers, there is data just beginning to emerge. One of the recent ones shown that to a large extent, different assays were interchangeable, and good news is that they all performed quite well when you compared to, let's say, age and sex, or age and sex and ApoE, which means the biomarkers have good value. However, there's a lot more that we need to do. There's a lot more data that need to be produced. I'll finish off with clinical trials, how p-tau may be used in clinical trials. And this is just a simple analysis of how useful p-tau could be to clinical trial recruitment and retention. So for example, and these are data from ADNI if you were to just use PET scans, A-beta PET scan to recruit 1,000 individuals hypothetically, and assuming that there's 20% positivity rate, in a very simplistic form, you have to recruit 5,000. And that if you have a modest cost of $3,000, then it will cost a lot. But also there will be a lot of logistical challenges, as we know. But then we propose that if you use blood tests and then you combine with PET scans, then you use a blood test to screen. Because what we did find was that there was about 40% of those who were A-beta positive in ADNI who were also phosphatide positive. So if you assume this, then you could just screen 5,000, the same 5,000 with blood, which is much simpler. And then the 2,000 who would show A-beta positivity, you could then take them extra to do A-beta per confirmation, which simplifies everything. But then there are some key things we have to think about, because I know phosphatide is being used in clinical trials now, that therapies target amyloid and tau. Those are the ones that we would focus on. But then if p-tau is increased when you have A+, and then they further increase when you have T+, then, and also this study suggested that they even mediate this relationship. Could that then affect the use of p-tau as an outcome measure when you're going after tau, or if you're going after amyloid in a clinical trial? And there are some considerations that we have to look at, and I'll just cover these in the next slides. That probably we should use in conjunction with other biomarkers, for example, NFL, which is already used, and then the plasma A-beta as well, the IPMS methods. And that could there be a possibility of being able to develop markers that are specific enough for amyloid and tau separately? As in, if we know that the two schools of thought that you could have the amyloid cascade hypothesis where tau A-beta starts, amyloid starts, and then induces tau phosphorylation, and then there could be potentially other non-amyloid dependent pathways. Could you be able to separate them? At this point, we do not know, but there could be some ways. And then could they also develop, let's say, for example, an aggregation? Because if tau PET and tau positivity based on brain aggregates is based on what is happening over time in terms of aggregation. That's what tau PET does. Could we then try some markers, for example, that are in blood or CSF as aggregation, as T biomarkers? And here there's one marker that I think could stand a chance, tau 368, which is the end of the micro to go bind the region of tau, and where you have the levels, if you take as a ratio with total tau, it tends to drop over time. And that when you look at z-scores and trying to look at the changes over time as a function of A-beta scintillate, then this CSF 368, tau 368, which is not a phospho, it is a fragment, it comes quite later. It comes later after A-betas and phospho-tau. So could it then be that this could be a better measure of T in terms of from a biofluid perspective? And that we do not know that when amyloid clears out of the brain, what happens to CSF and plasma p-tau, especially for plasma? We don't know. So these are things that we also have to look at that in plasma, for example. And yeah, exactly the same way. And that could we use, for example, the phosphatau A-beta ratio as a better measure of fibrillar A-beta. So in conclusion, blood biomarkers are pretty promising, and p-tau being the new addition, but there's a lot that we need to do in terms of validating to enable clinical use, and also how to adapt to different contexts. And there's several questions, and I'm sure there's several other things that I did not even think about or could include in this talk. but there are several things we have to do in terms of defining what the meaning of abnormality is, and then how to establish confounding factors and what increased p-tau means and what does affect clinical decision and how we could validate against different biomarkers. I'd like to thank everyone, other people that I've been working with and collaborating with. Thank you very much.
Oscar, I believe you're muted.
Sorry, I lost the voice.
Thank you, Thomas. It was an excellent presentation. When I thought that I knew everything about biomarkers, plasma biomarkers, then came your presentation. And it was very, very informative. We have a few questions. Barry Greenberg, wonderful talk, Thomas. It is known whether circadian rhythm effects may impact on plasma biomarker measures.
At present, I would say we do not know, but I know of a study that we are going to do that will provide evidence of this. So maybe we should hold on for just a short while. Okay, Gil Rabinovich, wonderful talk, Thomas. A couple of questions. Some studies suggest longitudinal decreases in CSF p-tau as disease progresses. Are there indications that plasma p-tau follows the same trajectory? How might this impact diagnostic performance? For example, will it lead to false negatives in dementia patients? Do you see value in looking at panels of p-tau at different epitopes for diagnosis or disease staging? Value combining A-beta and p-tau measures versus either alone? Okay, so that's several questions in one. Yeah. First thing, yes, CSF p-tau decreases over time. We've seen similar for plasma to an extent. I think we saw in this ACTA paper from us, and also I think in this Nature Medicine paper from Lund with Janellis as the first author, they've shown something to an extent. But there's a lot more, and I agree that we don't have enough longitudinal data at present. I think this would happen, and then how could that affect the performance? Well, because if we have someone who is quite well into the dementia stage, then it means cognitively there will be a high suspicion of AD to an extent, if it agrees with the clinical workup. But then if you see CSF and plasma both show negative, then that could be a problem, yes. So that could be where one advantage is that PET has, for example, because that looks at the other way, where you have buildup over time. So yes, that could have a problem. But saying so, the changes are not massive. From what we've seen, the drops over time, it kind of goes just quite uniform. It's not like a drastic drop in the levels, because you get to see changes. Maybe it's due to degeneration of neurons or neuronal loss. And then value in looking at panels. Yes, that, for example, we could see, and what's quite nicely shown in this Nature Medicine paper from the Bateman group, that in familial AD, you get to see different phosphatones that sort of change at different stages of the disease. So it seems like a baton, one gives into the other nicely, but there's still some overlap. So that could be quite important. However, I also see reason in that could also cause some confusion clinically, because if you give a result to a clinician and then you've done a panel, let's say a panel of 3 phosphatals, one is positive, the other 2 are negative, then that could make it more difficult to interpret. And then the value combining A-beta and plasma versus alone. I think the phosphatal A-beta ratio comes in quite nicely, especially from CSF. I think it's like it provides a very good measure of clear-cut AD, I think, compared to if you just AD or CSF phosphatal alone. And have there been attempts to measure with blood spots or delayed signification for remote collection in p-tau? I think there have been some studies in CSF, but not in plasma. We are now trying. There's a PhD student who's trying to transition the work that I showed for NFL into phosphatal. And role does the blood-brain barrier play in regulating levels? Yes, this is one topic that we're also very interested in trying to understand, that we don't know. That's the simplest thing to say, but we have our own suspicions that potentially if there's a massive disruption of the BBB, then you will have a lot of changes happening with like massive changes of p-tau coming into blood. But however, from what we've seen, that tends to clear quickly over the next several days. And then from Ranjan, I wonder whether you have thought about including a measure of cognitive reserve assessment.
So we have Susan Kraft. She said, have there been attempts to measure p-tau or A-beta with blood spot or delay centrifugation to allow for remote collection?
Yes, so I think I've tried to answer that already. Sorry, Oscar, I was going ahead of
Okay.
you. And then I tried to Ted's
so
also.
Ted Wilson, fantastic presentation, Thomas. What role does the blood-brain barrier? Oh, you said the blood-brain barrier regulation. That's okay. Rajan Dwara, great talk, Thomas. I wonder whether you have thought about including a measure of cognitive reserve into your assessment of correlation of p-tau to the clinical status or preclinical or early MCI cases.
I think, like, for example, with the recent paper from Biofinder, where they show that you could use an algorithm to predict, we're getting there gradually to integrate p-tau and cognitive changes, also clinical data.
Okay, well, I think that we are going to conclude here earlier than we expected.
Thank you.
I would like to thank you all the presenters. This was an excellent morning. We hope that you have an in-person meeting in the spring of 2022. More details to follow. Nina will keep us informed. And now now you can take it from here, Hannah.
All right. Yes, I just want to echo Oscar in saying thank you, and we hope to see you all again in the spring in person for our next director session. But wow, it is crazy, just the wealth of information that can be shared in this Zoom format, and really great job to all of our speakers today.